Yeah, thank you.
Yeah, just hand it back to me.
All good. All right. Welcome, welcome, welcome. So happy to see everyone today. We're about to get started. Thank you all very much for coming out on this beautiful, slightly sprinkly day. And welcome to the 2025 IEP workshop. We are your co-chairs on the organizing committee. I'm Rosemary Hartman from Department of Water Resources.
I'm Adam Nanninga with the U.S. Fish and Wildlife Service.
And yeah, we have an awesome program for you today. I have heard morale is a little bit low. It's a really tough time for a lot of folks, especially our federal partners for sure. So I'm gonna do my best to lift it up a little bit. I can't do anything about the horrible political situation that we're currently in, but I do know that a lot of people are not feeling appreciated at a number of levels. And what I can say for sure is that here in this room and in the community that is online right now, we appreciate you. You are appreciated. You do amazing work, and we will have to see what happens in the future, but we do have a really great community here within IEP. And I have nothing that will fix this, but I have some good puns. Because I am very glad to be here. We're gonna hear a lot about a lot of cool science. And if you are feeling a little crabby this morning, I can suggest a really good anchor management class. Or just coffee. We have coffee in the lobby. Coffee is sponsored by ICF. Thank you so much for the coffee. We can't pay for our own food and drinks, but we have wonderful partners that can. Please, though, no food or drink in the auditorium. If you are carrying a travel mug and sipping out of it, I'm going to assume that's filled with water. You can have water in here. Please don't do anything to prove me wrong. Yeah. So logistics, we are here in the auditorium. In the event of emergency requiring building evacuation, please remain calm, take your belongings with you, and proceed to the nearest exit. We'll have a safety relocation point across the street on 1615 9th Street, which is Franklin D. Roosevelt Park. Look for building staff holding the first and second floor signs. If you need any help navigating the building or any special accommodations, you need listening devices or access to a lactation room or help getting to the second floor because you do need a building badge to use the elevators, please see Christine out of the registration desk or myself. We can get you set up. Coffee is right outside. You probably found that already. Bathrooms are just around the corner, kind of under the stairwell. There is a food court across the way. There's also a variety of local lunch options in the little area guide that you can access on the website with a QR code. But there's some food right there. And if you are participating in the mentor lunch, we have food provided for you. And I just ask that you meet over around the other side of the building near the security desk, and we'll walk you upstairs to the rooms that we have reserved there. Few other things. This is being recorded and broadcast via Zoom. So as you're asking your questions, if there's something that you don't want the world to hear, maybe save that for a private meeting in follow-up. There is free Wi-Fi. It's CNRA guest. It's open. You just have to click a little accept terms and conditions thing, and you can get online. But I'm sure none of you is going to be multitasking during this meeting because it's going to be so awesome. Also, a little plug, there is going to be a poetry slam before the first poster session this afternoon. This is a little chance for people to plug their posters in the form of a short verse, song, or skit. Even if you don't have a poster plug, but have a poem about sturgeon you want to share, please do. And we'll just line up here before that happens this afternoon. After hours today, Cal Neva is having a happy hour over at Drake's the Barn, across the river in West Sacramento. And tomorrow we're having a little after hours hangout over at Pipeworks climbing gym. If you climb or want to try climbing for the first time, a bunch of us will be going over there. Or if you're not up for climbing, you can come with me right next door to the bar and have a beer or other beverage. Yeah. And there's food. Thank you, Pete. Talk to Pete if you need more information. And there's a flyer on the QR code out front. So a huge thanks to the planning committee, especially Christine, Job, and Stephanie Fong. We could not have done this without you. You do all the hard work, and Adam and I get all the credit. The rest of the planning committee, the CDFW IT staff who are helping make sure that this whole thing runs seamlessly, and the huge number of volunteers we have helping keep time and run microphones. The natural resources volunteer group who's helping with the registration so that we can all be in here. Yeah. And with that, I think we're ready for our next talk. Stephanie, just let me know when you're ready.
Thanks, Rosie. Can you guys hear me? Okay. All righty. So welcome everyone. It's been a little while since I've gotten in front of you guys and addressed the IEP community. So for those of you that don't know me, my name is Stephanie Fong and I'm the program manager for IEP. And I know a lot of people often wonder what it means to be part of IEP or what IEP is. And maybe this will help a little bit. So IEP is not an agency and it's not an entity. It's really a community. And it started with 4 agencies who recognized the need to perform studies to help them really gain a thorough understanding of the requirements of fish and wildlife resources. And beyond the signatories of our MOU, we have a huge network of incredible partners. We have the Delta Stewardship Council, who not only participated in and still does in core IEP teams, but they funded the Delta Independent Science Board's programmatic review of IEP. And they supported Steve Culberson's time as our IEP lead scientist. We have many partners at UC Davis, like the Otolith Geochemistry and Fish Ecology Lab. And, you know, these guys have been working to study, reconstruct the life history of many of the important fish species that we care about in this estuary. Also at UC Davis is the SMELT team from their genomic variation lab. And they not only develop new ecological monitoring tools and research management applications, but they also are our main champions for ensuring the genetic robustness of our Delta smelt at the cultured Delta smelt at the FCCL. So they work really closely with the UC Davis Fish Conservation and Culture Lab. And these folks have been actually maintaining and culturing Delta smelt since 1992. I think right around the time some of you guys were probably born. Not me. And they also work really closely with the U.S. Fish and Wildlife Services at Livingston Stone National Hatchery, where our refuge population resides. We even have really close partners in the private industry, like ICF on cue. Jason walks down the aisle, who not only provided the coffee and tea bar for us today that you'll be enjoying over the next few days, but they've provided novel research as well as more traditional monitoring of some of our very special key species. As have folks at Kramer Fish Sciences, you'll see them about as well. And I just have to say, I love this picture of the shallow habitat sampling platform as it's kind of motoring through. So thanks to Joe for sending that over. We also have affiliations and partnerships with groups like the San Francisco Estuary Institute, QEMF, and the CalNEVA chapter of AFS. And these groups provide expertise and foundations that we can really build from to strengthen our science. Together, we make up the IEP community. And it's the unity and community that keeps us tight and united. And I have to say, I was trying to go to sleep a couple of nights ago, and Rizzo was on TV and said this, and I immediately popped out of bed just to write this down. So as a community, we kept performing high quality science through 55 years of ups and downs of funding, ups and downs of changes in political structure, everything. Some of you may want to ask some of your colleagues who've been around a little bit longer about the CalFed days or some of the other funding opportunities we've had along the way. But we've also identified and addressed environmental challenges like the pelagic organism decline. We've collaborated on our shared goals to achieve commitments that drive us forward as a scientific community. And because we work together, we built really great relationships that have helped us find solutions for challenges that were put before us. So as I was preparing for this talk, sometime around 9 last night, I started looking at the first talk I made for folks at my first year as the IEP program manager back in 2019. And these were some of the things that we were starting to address. We were struggling with an unreliable fleet and missing some sampling because of it. We were looking for new technologies to help us answer more modern environmental questions. And we were struggling to publish our data in a timely manner. We were struggling to do the analysis even that was needed to answer the new modern questions we were having. So now we have a more reliable fleet. It's actually larger than it was before. And because we share resources across agencies, we can help ensure each other's monitoring and other needs are covered. We've developed new technologies beyond those that are listed on this slide, and we're making great strides with things like Sherlock. We're publishing data on the Environmental Data Initiative more often and faster than ever before. And we have a greater standardization, so our databases and data sets can be compiled and shared into larger data packages that are more useful for everyone, like Zooper or Delta Fish. And we've led trainings on things like metadata and data stewardship and coding in R and statistics to promote better data usage. Not to mention all the various reviews that we've performed and journal articles that we've published, because it's really an astonishing amount of information. Just in Steve Culberson's bibliography for 2024, there were 75 publications built from IEP data and our surrounding resources. 75 publications in 1 year. It's pretty amazing. So as I think about our current environment and I listen to the concerns of the folks that have been talking to me recently, I wanted to provide some things for you to maybe consider. I think maybe we should be trying to really pay attention to the meaningful information. So slow down and take a moment to think through some of the broader considerations before we react. Sometimes things aren't as bad as we think that they are initially. We just need a minute to think it through. Hear each other, not just listen, but actually hear each other. So we get it when someone else needs some help, and offer that help when we can. Celebrate the positives, no matter how small. Carl Wilcox is here today, so that's like awesome for me. So I'm going to celebrate that. Greg Erickson, who was my predecessor, is maybe going to make a surprise appearance one of these 3 days as well. So keep an eye out for him. And then take care of yourself. Don't fixate on trying to predict what might happen. Control what you can. Look at things around you, and ask for help. So if you're taking care of yourself and you're asking for help, someone might just have what it is that you need or might have access to that help that you need. I'm also reminded of my 4 favorite takeaways from Steve Culberson's 9 things we should be discussing as a community. This citation to the paper is down below. So 1 was constant dialogue is fundamental. And I think this is not only true, but things have been changing daily and sometimes hourly in recent months. So checking in with your partners is really needed now more than ever. 2 was near-term knowledge requires long-term context. And so we have to remember to keep an eye on the horizon to kind of help steady us as we go through this ebb and flow of tides. The 4th thing that he mentioned was cooperation is what leads to success of multiple agency efforts. So the charges of our various organizations are a little different, but they do touch on one another. And for those areas that they do touch on, we can look for efficiencies that benefit us. And those efficiencies will helpfully help us overcome things faster and easier than they would have if we were working at them individually. And then people matter and their feelings matter. So lifting each other up instead of climbing over each other is an important concept to foster. Looking for leaders and not just looking up the chain for leaders, but down and across the sides, right? Because they can be all around us and we can lead even from the very initial early careers positions that you guys are filling, right? And you can be a good leader through example from every single level, and it'll all help us grow and learn faster. And please lead with empathy and gratitude. So in that vein, I am grateful for the relationships that IEP has created for all of us to be able to do things like take the experimental release of Delta smelt head on. This was a really difficult situation where we were trying really hard to quickly start releasing cultured Delta smelt into the wild with very little information about what we knew we could and could not do and still have these fish survive. But even though it was a really huge feat, we were able to carry it out with a big group full of IEP scientists and IEP fleet operators, because the IEP community consistently strives to look for solutions and implements them with the resources that we have available. So for many reasons, I am profoundly thankful. Thankful for all the teams who work together in our community to collect the data summarize and make the data available, integrate the data, and, take it from multiple sources to perform really thorough analyses, and turn that data into real information that our managers can use. And thank you for showing up every day and doing your best, because sometimes that's all we can do. So don't forget small successes. And even though these are all sort of field photos and just one lab photo, my sentiments absolutely also include the dozens and dozens of IEP scientists who are working in front of their computers as well. So with that, I just wanted to thank you all as members of this IEP community. Hopefully we can bring up the mood. And with that, I'm gonna give it over to Karrigan, because I think he might be able to use some of my extra time.
I am thrilled to introduce Dr. Karrigan Bork from the UC Davis Center for Environmental Law and Policy and the Center for Watershed Sciences. I'll admit, when we thought it would be a great idea to have Dr. Bork give us a little mini training plenary talk on the law of the Delta, it seemed like a good idea at the time. In our current climate, I think it's an even better idea to get us a grounding on the laws that make water in the Delta work. So with that, give it over to Karrigan.
Great, thank you. Appreciate the introduction. I'm super excited to be here. It's Bork like work, not Bork like dork, which is what my mom has always said, and I find useful, but I should have told you. No worries. Yeah, so I'm a professor at UC Davis Law School. I teach water law, environmental law. I teach property. I promise I will not cold call you, so no Socratic method this morning, but I would appreciate it if you stayed awake. That's my bar for my students. So if we can hit that, we'll be great. So we're going to talk today about the law of the Delta, and we're going to talk at a pretty high level to kind of get an overview for all the laws that are at play in the Delta. This is a reference to the law of the river, which is this term coined to describe the huge mess of law around the Colorado River. You've got a bunch of different states, you've got international laws, you've got state, and you've got Federal Endangered Species Act laws that are in play there. And it's seen as this complicated thing that no one can fully understand. And I think the Delta really falls into the same place, even on the legal side. And so I just want to acknowledge that up front. There's 100 reported court decisions that deal with the Delta, tons and tons of water board decisions. Literally every time I talk about this, I learn something new. And I've been doing it for a little while now. So, I think we go into it with that idea that we're kind of getting this high level overview and there's always more to learn in the details. It's probably a good way to approach this. So I'm going to start with a little overview of California water rights. So kind of a water rights 101 piece, walk through the major forms of water rights. Then we'll move into restrictions on water use. So first, how you get the right to use water, and then how we restrict the right to use that water. We'll talk a little bit about Bureau of Reclamation, the Central Valley Project itself, and then we'll talk about water flows in the Delta. And you all have backgrounds probably in a lot of these different pieces, and you'll see ways it fits together. And I'm going to try and build in the law side of that. I'm leaving out a whole lot of the law here. So we're not talking about the Corps of Engineers and the levees and the flood flows and the operational curves, kind of all those aspects. We're not talking about River and Harbors Acts. We're not talking about FERC, not talking about the tribal treaties. So there's a whole bunch of law that we just don't even have time to kind of cram into our hour today. And I'm hoping to leave some time at the end because it is really fluid right now. Things have been moving around quite a bit on the water side, and I want to leave time to take questions from you all about that. I'm not sure that I have answers to any of those questions, but we can ask them and think about them a little bit and try and get a feel for where we're heading. And again, this covers a huge space. This isn't new to you, right? But we're talking about law that covers just an enormous chunk of California, which means the laws and the places that we impact when we think about how water flows through the Delta is really huge. So we've got a lot of different kinds of water rights in California. Water rights are a state property law issue, and so states come up with their own systems to allocate water rights. The way we do water rights in California is very different than the way they do them in Nevada or Oregon or Washington or Kansas or over on the East Coast. Right, so this is determined at the state level, and state law generally controls who gets what right to use what water. And then we have a federal overlay on top of that we'll talk about because federal law tends to win out over state law when there's conflicts. Not always, and we'll talk about some of those exceptions, but we start with the state law piece and then we lay the federal law on top of that because state law determines who has what property rights in the water. So the kinds of water rights I want to talk about are riparian rights, appropriative rights, area of origin protections, which are kind of water rights, kind of not, and contractual water entitlements, which are definitely not water rights in California. In other states, they're seen as something much more akin to water rights, but which are important for us for thinking about who has the ability to use water. What we're leaving out then is groundwater rights. So Sigma and all the rights to use groundwater, which is a big deal, of course, on the Sacramento River and for the Delta. You're looking at like almost 1 million acre feet are the estimates I've seen of water that comes out of the Sacramento River and is used as groundwater, but we're gonna like push that off. Prescriptive water rights, which is when you basically steal somebody else's water for long enough that the state says, yeah, that's fine, just keep it. It's fun, it's really interesting. It's not very important, unfortunately, so we're not going to talk about that. Federal Reserve water rights, these are the rights that come along with federal land when the federal government reserves land. I'm not going to talk about those. Tribal water rights tend to be a piece of that. Those really aren't very much at play in California's Delta because of the history of not honoring the treaties originally signed with the tribes in the Central Valley. Since those tribes don't have treaty rights, they tend not to have big reservations and big water rights associated with that. So we aren't going to talk about those. And Pueblo rights, which are a really old kind of Mexican water right associated with cities in Southern California. Only 2 cities have them. Again, not really in play for us. So we're going to focus on those first 4 kinds. So riparian water rights came from the East Coast. When California became a state, we've got the California legislature looking around thinking we've got to pass laws. Like we probably need laws for the state. Like murder should be illegal and stealing stuff should be illegal. Where do we go to get that? That's a lot of laws to pass all at once. You think about like ginning up the whole set of laws for the state. And so what the state did was what a lot of states did at that point and brought in English common law. And so California said, whatever the rule is in England, we'll make that our rules here. And then we'll like do tweaks to that. Anything we change to get away from English common law will take precedence and be more important and we'll follow that instead of English common law. But if we don't pass something, then that's where we can pull our law from. That's where most of the books that had laws written down in them were coming from at that point. That's what people tended to be trained on. So it made a lot of sense. So that's April 13th, 1850. California legislature says English common law applies in California. And the English form of water rights were riparian rights. So these are rights that you get by owning property. So if you own property that's riparian, meaning if you own property that butts up against a water source, you've got a right to use water from that water source on that property. Okay? So riparian right, gained by property ownership, not gained by use, not lost by disuse, the way that we think about other kinds of water rights. You just got it by being there. These water rights didn't go away. If you've never used them, if you have riparian land, you could decide tomorrow to start pulling water out of the river and putting it onto your property. The use does have to be reported to the State Water Board. That's a newer thing we've added on. Historically, wasn't part of that. There's all kinds of things we can get into about how good a job we actually do of getting that data reported to the Water Board. Surprisingly good in the Delta. It's actually a spot where the Delta is a leader for the state. I think in the Delta, the numbers I've seen talking to the Delta Water Master are like 90, 92%. Other areas in the state, you're at like 50%, 75, even lower in some areas. So it's supposed to be reported, but a lot of people don't, and you don't lose them if you don't report them. And these rights don't give you a right to a set amount of water. They're correlative rights. So your right is correlated with all the other riparians in a system. You can think about the water in the river as a pie, and all the riparians get a slice of that pie. So in a big water year, the pie is really big, and everybody gets a bigger slice. In a water year where there's not as much water, that pie shrinks, and everybody gets a littler slice, but everybody's still getting a little bit of pie. Right? So these water rights are correlated with other riparian right holders, and the kind of way we define how much water you get, how much you can pull out and put onto your land, is what you can reasonably use on that land, given a reasonable method of diversion, reasonable method of use, and a reasonable purpose of use of that water on your land. There are a lot of limits to these water rights. But these came from England. These are the water rights that are still used in most of the eastern part of the United States. But they evolved in rainy England, right? It's a little bit different than California, much wetter place, much wetter environment. And so there's a lot of aspects of these rights that make them a pretty bad fit for California. The first one is land limits. So you have to own the land. You can't just be using the land. And if you think about the miners, the people who were kind of first worrying about how much water they could get and were first having fights over water, they didn't own that land. They were mining state land and federal land. They wanted to get in on public land, get the gold out, and get gone as quickly as possible, right? No interest in actually taking ownership of that land. And that meant they didn't have riparian rights. So made it pretty hard for them to say, this water is mine. You can't take it. I get to use this water to blast this hillside with my water cannon and do the hydraulic mining thing, right? So riparian rights, not a great fit for those miners. You also have to use the water within the water course that you take it from. So you can't transport that water over long distances. You have to use it on land that's contiguous, that's riparian to a waterway. So if you own land that didn't touch a river, you didn't get a riparian right to it. In the eastern US, no big deal, right? It rains there, it rains all summer. You could still grow crops on that land. Out here, if you have land in the Central Valley that doesn't butt up against a water course and you can't get any water for it during the summer, that land's not worth a whole lot, right? So pretty poor fit for the California system. They're also not transferable, so you can't sell your water right to somebody else. We tend to think it's important to be able to sell property to other people. You couldn't store these water rights for more than 30 days, so you could temporarily store them, but 30 days isn't going to get you through a California summer, let alone one California year to the next California year where you might want to save a big water year for a low water year that you know is coming. And last, you're limited to the natural flow of the river. So you can't use imported water into a system. You probably can't. You definitely can't use stored water post-release. If somebody downstream claims it, it's not clear if you're kind of at the end of the system, if you can use it. Courts tend to say no on that. And what we generally define that natural flow as the unimpaired flow of the river. The probability in there reminds me, as I talk through this stuff, I mean, I definitely have perspectives on the law, what I think the law should be, but I'm trying just to tell you what the law is. And any spots where I think it's really unclear or where I think people will disagree with what I'm saying, I'll caveat, there we go. I'll warn you. So you know that it's not entirely settled in those areas. Okay, so we've got these riparian rights, not a great fit for California. And so the miners, as I said, were the first ones really fighting over this, and they established a form of water rights that's a lot like what they used for mining claims. And these were called appropriative rights. So just like a mining claim, you got this appropriative right by going out and starting to use the water. You didn't have to tell anybody. You didn't have to post notice anywhere that you were going to do it. You just diverted the water and put that water to some use that was not wasteful, right? Some beneficial use. So pre-1914, that was it. You divert that water, boom, you got a water right. And those water rights are still good rights. So if you've got somebody who started diverting water in 1855, that water right is still a good right today. So they got that appropriative right just by diverting, not by telling anybody. There was no analysis. There was no like, what will this do to the fish in the river? Just not a concern. And so whatever water was there could be grabbed up with those rights. After 1914, you did have to apply to it was an early version of the water board for a water right permit, and then eventually you get a license. Those rights are limited to the amount in the permit, but those pre-1914 rights are based on the amount that was used and has been used historically for that water right. This gets really tricky. If you're saying, hey, I got this water right in 1860, you got to show how much water you were using in 1860, and then that you've used that water every year or with a break of no more than 5 years ever since you started using that water. Right? And it really matters because the way we figure out who gets their appropriative right first, these aren't correlative the way that those riparian rights are. It isn't like all the appropriative right holders get some water every year. These are based on first in time, first in right principles. So the oldest water right gets their water first before more junior rights get the water. And that means if you've got an 1860 water right, it's really valuable. right? It's really strong. You're much more likely than everybody else in the system to get your right. The trick is you have to be able to prove up that right. So those older rights are a little bit weaker in terms of being able to prove them up and really show that you have them. But if you have them, right, you do get a much stronger right that you're able to keep other people from using. All right, so some of the things that make these better fit for California, land ownership was not required, just use of the water. Storage is allowed. You can store this for longer time periods. Transfers are allowed, including transferring the water out of the watershed. And the quantities in some ways are more knowable and more stable than what you'd have under a riparian right. Right, if you have claimed a water right on the system and you have a very old water right, you know that in most years you're going to get that water. Some new user coming in and saying, hey, I actually want to use some water too, they have to get in line behind you. And that's really different than in the riparian system where another riparian can come in and say, hey, I'm going to start using water too. It's going to cost you some water because we've got to share this because these rights are correlated, right? So in some ways, this gives you a more definite water right. These water rights spread really quickly throughout the western part of the United States after California developed these water rights. So they just like took off, right? Because California isn't the only state that has this kind of weird hydrology, weird climate, where the riparian rights weren't a good fit. But California kept the riparian rights around. All the other states got rid of them that are in the western US, but California said, no, we like these riparian rights, probably because of some political stuff, because these tended to go with really big landowners who had money and power and have been in California for a long time. There were a lot of efforts to get rid of riparian rights, and the courts rejected them pretty consistently. So these riparian rights stuck around. And so now we still have riparian rights. We also have appropriative rights. And the appropriative rights lose to riparian rights in times of shortage. So riparian users get their water first. Appropriate users are in line behind those, and that's based on really clear state law and a lot of precedent. And these appropriative rights, unlike the riparian rights, because they're permitted, if you want to change your place of use or your purpose of use, you have to go and ask the water board for permission to do that. If you have an older water right, a pre-1914 water right, you have to at least let the water board know, and it gets kind of mushy how much permission you have to get for those older rights. But the time of use is actually fixed based on the water board's reading. There's some disagreement among scholars whether that's really true, but the water board sees these water rights as fixed at a particular time. Some of them are fixed at nighttime because somebody started diverting in 1865 and they were only pulling water out of the river when it was dark. And that gave them a water right to use X amount of flow in the dark. And that's still the restriction on those water rights, right? So time of use, normally seasons, but can even be time of day restrictions on water rights. Okay, so those are our major kinds, riparian rights and appropriative rights. These area of origin protections came in later, and they were a way to address people's concerns about big water projects taking their water and moving them to the other areas of the state. Right, so you think about something like Owens Valley, this produced a lot of this concern, really made people worried because you had a valley that had farming, that had trees, and then Los Angeles came in and took that water, shipped it all down to LA, and if you've been to Owens Valley, it doesn't have all the trees and the farming that it historically had, right? Very different place, and the people who were there were worried, and it made people everywhere else in the state worried that similar things would happen to them, and so we get these area of origin protections that protect origin watersheds for big water projects from the negative potential impacts of those water projects. So the most important one for us is the Delta Protection Act. It came in 1959, about the same time our state water project was authorized, and this limits water available for export from the Delta in order to protect water quality and water use in the Delta. Right, so this is where the big concern with salinity kind of comes in here. There'd been some concern about salinity already, but this is where you get a lot of protection for it. And the water board has a lot of power to protect this in Delta water use and in Delta water quality. There's some language there from a California appellate court case talking about how much power the board has to protect these interests. These show up in other areas. There's San Joaquin River ones. There's some up in the northern part of the state. So not limited to the Delta, but for our purposes, that's the most important part. And as you know, the Delta's got a ton of water use that is actually in Delta, right? So we've got a lot of farmland in Delta, a lot of urban areas in the Delta, and salinity is a big deal in the Delta. And I think you all probably know this already. This is a pretty famous chart, but it shows kind of before the water projects happened here on the left side, how far into the delta the salinity would come. And then on the right, you can see how successful the projects have been about limiting the inflow of saltwater into the delta. So the left side is 1921 to 1943, right side is 1944 to 1990. Right? Really have pushed that saltwater edge way farther out in the delta. So those water right holders in the delta, there's about 3,000 total diversions for in delta use. That's a lot of diversions. Those are pre-1914, post-1914, and riparians, and they're all covered by that Delta Protection Act. There's also a lot of water rights upstream of the delta, so on the rivers that flow down into the delta. And you'll see in some water years, those upstream users, those right holders farther up on the river, combination of appropriate and riparian users are pulling out almost the full flow of the river and really not leaving very much in the delta for in delta use or to keep that saltwater edge out. And in those years, the water projects are super important in terms of getting fresh water into the delta and keeping the saltwater from intruding. And then for the Central Valley Project and the State Water Project, they have 34 appropriative rights between them. They are all appropriative. They're not riparians because they don't actually own the land where that water is being used, right? They have the water and then they're selling it to people who use that water. And so all of their water is appropriative. Water is held under an appropriative water right. The terms of those appropriative water rights issued by the state allow for the reshaping of the rights, both for water quality and for environmental purposes. And then that water that's held by the Central Valley Project and the State Water Project is parceled out via contracts to all the contractors with those projects. State Water Project contractors, the State Water Project could deliver up to 4.17 million acre feet, generally an average of 2.4. Water use about 34% ag, 66% everything else. Contracts were passed or signed really in the 1960s. They've been amended and were recently re-upped through 2085. Central Valley Project, bigger, more capacity, about 9.5 million acre feet max, 7 million acre feet on average. About 5 million of that is for irrigation, 600,000 acre feet for municipal and industrial use, and 800,000 acre feet for environmental use. They've got a lot more of these contracts, so 270 water contracts or agreements. And then there's kind of differences among these contractor groups. So when the water board or when the Central Valley Project came in, there were already water rights on these rivers. Right? If you think about Bryant Dam when it was built, there were people downstream of that dam who were already using the water, who had riparian rights to the water in the river, who had appropriative rights to the water in the river. And they knew when that dam closed, that river was going to run dry. 60 miles, right, of the river was going to be dry there. And so those people, those landowners there or right holders there were having their property taken, their water rights taken by the federal government. And so the federal government had to come to an agreement with them in order to take their water rights, sometimes willingly, sometimes less willingly. But this is the same process, kind of condemnation that the government uses when it's widening a street or a highway, wants to take people's property. They'll negotiate with you and say, hey, can I borrow the front 10 feet of your lawn to make this road bigger? And if you agree, then they'll pay you for it. And if you don't, then you go to court, go through eminent domain, and you eventually lose that. But the court tells them how much they have to pay you for it. Right? This is a power we give to our government under the Constitution. Same thing happens with water rights. And so we have these different classes. We have the settlement contractors who are both north and south of the Delta, and they gave up some of their water rights in a settlement agreement and then got water contracts. And their water contracts are essentially given a higher priority by the CBP. So they're more likely to get their water delivered because they gave up the water rights they already had. Right, so that's what they traded to get these kind of better contracts than other people get. We've also got the San Joaquin River Exchange contractors who are south of the Delta, similar story for them. And then we have a whole bunch of water service contractors who are, as I said, not so special, just meaning that they don't have this priority. And so if you look at water deliveries, you're going to see higher numbers for the settlement and exchange contractors, lower numbers for the service contractors. It gets messy when CBP is trying to protect kind of the minimum level they need for safety purposes and health purposes in terms of delivery for municipal users, because that bumps them up a little bit. They get a little extra protection because we think that use is so important. But this is generally how we deal with those contracts under the CBP. And then, like we said, both of these projects have really high capacities they can deliver, but they tend not to be able to deliver that full amount in most years. And there's a whole bunch of reasons for that. One, probably the biggest, is California's hydrology, right? That's probably the fundamental law of the Delta, is that climate controls ultimately how much flow we have through the Delta. But there's a lot of other things that help to control that. And so that's what we're going to talk about now, is some of these restrictions on water use. So we got our water rights. People pretty good with those? All right, cool. So let's talk about the restrictions. These come from a couple places. They come from state law and from federal law. They come from both laws that the legislature passes in California, so state statutory law. They come from constitutional law. So California's constitution addresses water use. Most Western states have found water use to be important enough to put it into their constitution. And it comes from the public trust doctrine, which is not written down anywhere. It's kind of court-created doctrine. You can think about it as common law, but it's not quite common law. Law professors love to get into arguments about where it comes from, and we'll talk about it a little bit. And then we've got federal law on top of that, mostly statutory, although there are federal constitutional protections for property that could come into play. Tend not to very often in California water for a bunch of reasons that we can talk about in the Q&A if you want. So let's start with our Constitution. Article 10, Section 2 says the right to water does not and shall not extend to the waste or unreasonable use or unreasonable method of use, unreasonable method of use or unreasonable method of diversion of water. All water types, pre-1914 rights, post-1914 rights, riparian rights, groundwater rights, they're all subject to this constitutional limit. And this was in response to a California Supreme Court decision, Herminghaus, that said, oh, we're actually not going to make riparians be reasonable. The only people complaining about it are appropriators, which would have made it really hard to have any of the rim dams in California. And so California passed a constitutional amendment to say every water use has to be reasonable. And that's going to limit people's ability to use water. And this means that if you have rights to water, no matter how you have the right to that water, you never have a right to use that water unreasonably, right? So what's beneficial, what's considered reasonable under the Constitution can actually change over time. So what was reasonable 100 years ago when we had a whole lot of water, we didn't have as many competing uses, we didn't care as much about the environment, can now be unreasonable. And that means the amount of water that you're entitled to can actually change over time based on how reasonableness changes over time. The most famous example was especially among the water law students, was this woman who was a riparian who was using the full flood flows of the river to kill gophers on her property. Appropriator was trying to take some of that water, and she said, no, I need the whole flood flows to kill these gophers. You can't put in your dam, you can't take water out, because then I wouldn't be able to kill my gophers every spring. And the court said that is not a reasonable use. So your riparian right does not extend to flooding your property in order to kill gophers. You still have a riparian right, but you're not going to get that much water because that level of water use is unreasonable. So this changes over time, and we have to consider the kind of whole status of the state as we weigh this. These quotes are from a couple of different California decisions, court decisions, applying the reasonable use doctrine. And courts say that this use cannot be resolved in vacuo, like in a vacuum isolated from statewide considerations of transcendent importance. Paramount among these, we see the ever-increasing need for the conservation of water in this state, an inescapable reality of life. And so the Water Board has this power to determine that water use is unreasonable. Courts have this same power, and the legislature has the power to say what an unreasonable use of water is. The sign saying recycled water used here for the green areas next to the highway, that's in part because the legislature said if there's recycled water available, it's unreasonable to use fresh, clean water to water those areas. All right, so legislature can weigh in, the Water Board can weigh in, and courts can weigh in. Next big limit is the public trust doctrine. So the public trust doctrine is an ancient law, it goes back to Rome. And the idea behind the public trust doctrine is that there are certain resources, certain things that the government holds in trust for us, the people, the public of the state. In Rome, you had your emperor. In England, you had your king. When the states became states in the United States, they inherited that responsibility from the king. The first 13 colonies got it directly from the king. New states that came in got it from the federal government, and we receive it because we come in the same way that all the other states, the earlier states, joined the Union. So when California joined the Union, the state government took on this public trust responsibility. So the state is a trustee for us, the members of the public, to protect certain assets for us. One of those big assets is waterways. So shorelines, rivers, streams, lakes, those are all public trust assets. And the federal Supreme Court has said there's certain trust resources that the state just cannot give away. And it really drastically limited when the state can give away any of the trust resources. And that means that water amounts have to consider, when we're deciding to give somebody a water right, we have to consider protection of those trust resources. It also means, under a 1983 National Audubon decision from, I lost a lot of the picture there of Mono Lake, but from this big decision in 1983, National Audubon, the Mono Lake decision, the California Supreme Court said, well, past decisions that were made without considering the public trust don't bind the state. Those decisions can be revisited because the state never had the power to give away the state's trust resources. So when we do give away trust resources, the state has a continuing obligation and a continuing power to supervise the use of water that impacts those trust resources and can call that water back if needed in order to protect those trust resources. Right? There's a huge political layer on top of this that we're not touching, but that's the state power. And the Water Board, when acting pursuant to its reasonable use and public trust authority, has never seen its decision struck down by a court in California. So the Water Board has a lot of power to use these things. It's not just the Water Board. So the state agencies like the Water Board, the legislature can use this public trust authority. It's also a basis for suits against the state or against trust users. That's what happened in National Audubon. For people who followed along some of the lawsuits to rewater, like the Kern River down in Bakersfield, to the lawsuit that led to the rewatering the San Joaquin after the 60-mile stretch of the river went dry. Those were private lawsuits that were filed based on public trust standing to actually enforce California state law. So the argument was, well, this state law is actually a legislative expression of the public trust. We, the beneficiaries of the trust, can sue in order to protect trust interests. So there's probably power for us to sue to enforce these state laws that are designed to protect the public trust. Federal courts and the state courts have signed off on that. So we have broad standing for us, the public, to rein in what people are doing, what water users are doing with their water, if it's having too much of an impact on public trust. And then this last one gets kind of technical. I mentioned, right, that the federal government has constitutional protections for property. So the government can't come in and take that 10 feet of your front yard to run the road through without paying you for it. When we go to the water setting, it's a little bit different, because if it turns out that the government needs to take part of your water right back to protect the public trust, then under that precedent we talked about, you never really had the right to that water anyway. The state made an error when it gave that water to you. And so the state doesn't have to pay you for that water that you're losing. Very clear decisions from the California Supreme Court on this, National Audubon. And then it's shown up again and again since then, and all the appellate courts have looked at it and said the same thing. There are people who are like, that's my property, you have to pay me for it, just to like fully disclose that, but they are wrong. And they lose when they go to court and make that argument. So this is not me like on cloud nine trying to tell you what the law should be. All right, so those are our kind of two big background pieces. Next one we want to think about is the 1969 Porter-Cologne Water Quality Control Act. This is the state version of the Clean Water Act. And this is really about water quality. Right? It's a water quality law. The State Water Resources Control Board is directed to attain the highest water quality, which is reasonable. There's that reasonable again. Considering total values involved, beneficial and detrimental, economic and social, tangible and intangible. So Water Board, consider everything, please, and figure out how clean our water should be. The way that this works is that the Water Board goes out and picks a stream where they're gonna, or a river or a delta, where they're gonna establish water quality rules. And then they figure out what beneficial uses should be made of that waterway. And I've been talking about the state water board. Of course, the regional boards do this in a lot of places too, right? For the Delta, we're talking about the state water board, and they figure out what the uses should be in that waterway. And once you establish those uses for the waterway, the next step is to set what we call water quality objectives. These are standards. At the federal level, they're called standards, but we call them at the state level water quality objectives, and that will allow for reasonable protection of those designated uses. Okay. And then you create a program of implementation that will let you achieve those objectives that you've set in order to protect the uses that you've picked. And those programs of implementation can be water right decisions, they can be water board orders, they can be done via regulation, lots of different ways to achieve those objectives you've set through your program of implementation. This is state law, but it's within a federal framework. So this is how California carries out its obligations under the Federal Clean Water Act. And so these regulations tend to be approved at the federal level. And there's also hooks at the federal level that say how often the water board has to update these things. We tend to, well, yeah, we never hit those deadlines. We're kind of constantly running behind, and periodically you'll have environmental groups that file suits to make the state get caught up. But this is the kind of general framework that we're looking at. And because it has that federal hook, we actually have the power as a state to make sure that federal actions that could impact water quality don't keep us from meeting the objectives that we've set. And so there's a kind of state veto over federal actions that are going to impact these water quality things. All right, so I said this is quality, but it's not just about quality. If you've been following the Bay Delta plan, you know that, right? It's often about flow, it's often about volume of water in order to support those designated uses. And federal courts have signed off on that and said that water quantity requirements as part of this can be part of your objectives. Like, that's okay, it doesn't violate the law, it's in the spirit of the federal and state versions of the Clean Water Act. So an example, you could look at Water Rights Order 90-5, and this enforces the water quality plan in the Sacramento River. So we have designated beneficial uses for that waterway. We have cold freshwater habitat, COLD, super helpful acronym, to tell us that's one of the uses that we're going to protect. Migration of aquatic organisms, spawning, reproduction, or early development. There's a whole bunch of other stuff, but I'm kind of a fish head, and so these are the things I keyed in on. And then the state, based on choosing those uses, and they choose those uses after consultation with the public, based on what the river has been used for, and based on the uses that people, the public, tell them they want to make of it, considering all those things the statute tells them they have to look at. They then set water quality objectives to achieve those beneficial uses. Right? And so one of the things that they set in those water quality objectives would be the location-based temperature requirements for protecting those uses of the Sacramento River. So how cold do we have to have it? Where do we have to have it be cold to create cold freshwater habitat and to create spawning, reproduction, or early development habitat to support those beneficial uses? Right? So use, objective, and then the program of implementation is this order, the Water Right Order 90-5. Also, the temperature control device that was added to Shasta was to achieve these temperatures. And then the downstream temperature controls that we manage Shasta to hit are part of these water quality objectives that are established under Porter Cologne. All good? Okay. Next, we've got the Delta Reform Act, the Delta Stewardship Council. There's a whole bunch of different kind of funding pieces and coordination pieces here. The big one I want to focus on is the last bullet. So there is a regulatory role. The Delta Stewardship Council writes that Delta plan, and they have appellate review of any action by a state or local agency that's going to impact the Delta in order to ensure consistency of those actions with the Delta. Right, so there is kind of a regulatory backstop there that shows up in the Delta Stewardship Council. There's a lot of pieces here, right? And it makes stuff super complicated to do in the Delta. And it means you can almost always sue over any decision in the Delta. You may not win, but you can get into court and you can stay in court for a long time. It's great for my students, not so great for management of the Delta. There's a bunch of other state requirements we don't really have time to talk about. I think it's really important to call out California does not have a set minimum flow law, right? There's no, we have to keep 40% of water in all rivers. We don't have any law like that. There's no set minimum amount that applies across all rivers everywhere, which means that figuring out how much flow is required is actually super piecemeal and really hard to do, and no one has done it yet. So it's something that I think is important to figure out, but it's hard to get there. We have the California Endangered Species Act, which in some ways is pretty similar to the Federal Endangered Species Act, California Environmental Quality Act, and then a bunch of other laws that apply in particular situations, which are sometimes relevant to the Delta, but we don't have time to go through kind of all of them right now. So we'll just keep moving. So our state level summary: public trust, reasonable use, Porter Cologne Water Quality Act, and then a bunch of additional laws that apply kind of in special circumstances or to particular species, particular rivers, particular places. Okay, so let's flip over then to the federal side. So we have the Endangered Species Act that applies for waters with listed species, National Environmental Policy Act, NEPA, which is the federal version of CEQA. We have FERC for non-federal hydropower and tribal fishing rights. Again, I'm not talking about those last two because we just don't really have time to hit them. And because they aren't super key in the Delta itself, although they're very important for a lot of those REM dams for the non-federal hydropower projects up there. So Endangered Species Act protections, we've got kind of 5 main sections. There's section 9, which is the core of the protection of the act, that says once a species is listed under the act, you cannot take that species. You cannot go out and kill that species. You can't harass it. You can't harm it. You can't look at it funny. Take is very broadly defined under the Federal Endangered Species Act. It includes even indirect harm via habitat modification. And that's gone up to the Supreme Court. The Supreme Court says that's fine. The old Supreme Court. I don't know what would happen now, to be fair. Okay, so section 9 prohibits take. Section 10 authorizes take. So if you get a scientific permit, you're doing that under section 10. If you have an experimental population of spring run Chinook in the San Joaquin River, that's going to be under section 10. Section 11, again, just like we saw with public trust, allows us, the public, to file lawsuits to enforce the Endangered Species Act. It can be timing elements if the federal government takes too long to review a listing petition. It can be substantive things like, hey, you made the wrong decision in that listing petition, right? So there's citizen suit provisions under section 11. Section 4 is how we decide what we're going to protect, how we designate critical habitat, how we write these recovery plans, which end up being not binding and super expensive, so they tend to be delayed a little bit. But there's a lot of species that are listed in the Delta, right? Our Central Valley spring run Chinook, the Sac River winter run, a whole bunch of species here. I put this up, and the last time I was presenting it, somebody was like, oh, it's a killer whale, how funny. That doesn't matter for the Delta. But of course it does, right? Because those salmon get eaten by the killer whales. And so we actually, if you look at the regulatory documents, they consider potential impacts to the killer whale populations too. That picture, I guess, that I showed of California should have like a big ocean bubble on it as well. So the piece of this that's probably most important for us to think about in terms of the Delta, especially given all the agencies in the room, is section 7. So section 7 of the Endangered Species Act says federal agencies considering an action must consult with the expert agencies, Fish and Wildlife Service or NIMFs, to ensure the proposed activity is not likely to jeopardize the continued existence of any listed species and will not result in destruction or adverse modification of its critical habitat. Federal action is very broad here, right? It could be something that a federal agency actually does, right? So operating the Central Valley Project. It could be issuing a permit. Corps of Engineers issuing a permit that's to someone who's going to destroy a wetland. That's a federal action. Federal government funding somebody. That's a federal action. Right. So this indirectly covers a lot of private actions and covers a lot of state actions. Great example for us, the Coordinated Operations Agreement, right? The way that we coordinate the State Water Project and the Federal Central Valley Project is a federal action, and so has to go through the Section 7 process. This is what we've seen kind of happen again and again and again, right? Over the last several years. So the way this consultation process works, we look first through informal consultation, sometimes with the development of a biological agreement. We ask, is a listed species likely to be affected by the federal action? If it's not, and the expert agency says, yeah, no, we don't think this action is at all gonna affect these listed species, good enough. You're done, you can go forward with your action. You've complied with Section 7 of the Endangered Species Act. But if it looks like the action will affect listed species, then you move into formal consultation and development of the biops, right? The biological opinions, and the expert agency for the relevant species carries that out, which is why we end up with 2 biops for the Delta, one by Fish and Wildlife Service, one by NIMS, dealing with the different species that they both cover. Okay, so the way the consultation process works, the expert agency determines whether the proposed action is going to place the species in jeopardy. Jeopardy is defined as if the action reasonably would be expected directly or indirectly to reduce appreciably the likelihood of both the survival and recovery of a listed species in the wild. So it's not about actually recovering the species. It's not about making things better for the species, right? The Section 7 consultation just says, is this going to force the species to extinction? The species numbers are going down like this. The agency doesn't have to turn and make them go up, right? They just have to make them not go down faster than they already were. So they can't be responsible for the extinction of the species. This is really an emergency room act, right? It's not an act necessarily aimed at recovery. You can incentivize recovery because the ESA is a big hammer, so you can get people to do things to make populations better. And you can get people to agree to do things through mitigation or other projects, but it's not inherently part of the Endangered Species Act, at least the way we're interpreting that act right now. There's arguments you can make that it should be, right? But that's not the way it's interpreted by courts right now. So you go through this biop and you have 2 possible outcomes. You can determine that the action is not likely to jeopardize the species. With that determination, the project proceeds with an incidental take statement. And that incidental take statement acts as your permit, both for the federal agency and for the parties that are actually carrying out an action to take those listed species that otherwise they couldn't take under the Federal Endangered Species Act. If the agency says, no, this is actually going to jeopardize the continued existence of the species, then the expert agency suggests reasonable and prudent alternatives to avoid jeopardy. And then the action agency can apply those alternatives. They can cancel the project and just not do it, or they can invoke the Endangered Species Committee, also known as the God Squad, and ask for an exception for the project. So not for the species, but for the project that you're looking at. Right, so it's not like, okay, you're going to let this species go extinct. It's more that we're going to let this project go forward, which may push the species to extinction, but what we're exempting is the project itself. So the God Squad, it doesn't just get to like wave their hands and make this determination. They're called the God Squad because the story goes, and it's a great story, so I don't really care if it's true. The God Squad was meeting at one point, and this hippie got up and he's like, man, you can't decide if these species get to exist. Only God can do that. And they're like, yeah, I guess we're the God Squad, because it's funny because it's sad, I guess. But the God Squad gets to make this call, but they don't get to just do it the way God would. They're constrained by the Endangered Species Act. So there's things that they have to find before they consent a project. They have to find that no reasonable and prudent alternatives to the agency action exist. They have to find the benefits of the action clearly outweigh the benefits of alternative courses of action that would be consistent with conserving the species or its habitat, that the action is in the public interest, that the action is of regional or national significance, and that neither the federal agency concerned nor the exemption applicant made irreversible or irretrievable commitment of resources ahead of time. That last one is super weird and I don't really understand it, so we won't worry about that too much. But when they make the decision, they also have to establish mitigation requirements. So what the project has to do in order to mitigate the impacts, even though they're letting the project proceed, knowing that jeopardy may result. Now, the members of the God Squad, and they also need a supermajority. It has to be a 5-2 decision among the 7 members. And the members of our God Squad, Secretary of Ag, Secretary of the Army, Chairman of the Council of Economic Advisors, the EPA Administrator, Secretary of the Interior, Administrator of NOAA, and then one person from any affected states in the project. All these folks generally designate people, right? They don't want to go and sit in these hearings and listen to the public talk about it. These hearings are public. It's only acted 3 times, so we talk a lot about the God Squad, but we've only seen it act 3 times. Tellico Dam, which is one of the very first big fights we had under the Endangered Species Act. This is the TVA v. Hill case about saving a little snail darter below a dam. And the dam was blocked and the God Squad met. The God Squad was created to address this. Like the legislature was like, this is crazy. We can't really care this much about a fish, right? The God Squad met and said, this dam's a terrible idea. And yeah, we should protect this fish, right? We should protect our snail darter. So they didn't grant an exception to Tellico Dam, and it only got built after a legislator snuck a piece in, a little rider into a budget bill saying that it would be authorized and would go forward. They met to consider Greylocks Dam in Wyoming, which had impacts on the whooping crane. They did grant an exemption there, but only after there was a settlement agreement. So they essentially just stamped the settlement agreement the parties in the underlying litigation had come to. And then they met as part of the Portland Audubon big fight about timber sales in the northern spotted owl habitat. And this is a big fight that spanned like 3 different presidential administrations. And they approved in that meeting 13 of their 44 timber sales. So this is something we talk about a lot. This is something that is relevant right now because there seems to be a move to try and set this up by the Trump administration. But there are rules about how it gets used. It takes a long time and historically has not been very successful. Past performance may not predict future results here, but that's what we've got kind of in the background. And then we've got NEPA, National Environmental Policy Act. This is why we have to do the environmental impact statements. So it's not a substantive law, it's kind of a look before we leap law. And that's it. So that's our restrictions on water use. California reasonable use doctrine, public trust, state statutory law, and federal law. And I'm running a little long, so we'll try and move along quickly. Putting this all together then, most of the rights in California are either first tranche rights, which is what I call like these oldest water rights, these pre-1940, 1914 water rights, or they're riparian water rights. And that means that the vast majority of water use in California is under rights that were granted pre-1914 or that you just get automatically. So no environmental review, no like considering if this is really in the public interest or not, no deciding if this is how we want to use our water or not. And then the vast majority fall either in that first tranche or second tranche of water rights. The second tranche water rights are the water rights that go all the way up to 1983 when we got the National Audubon decision. And again, most of those water rights were issued before we had modern environmental laws, were issued when the water board thought it had to approve requests for water if there was water in a stream. So they tended to always say yes, even if there were really negative environmental impacts, which means that the vast majority of our water rights were issued and are still being used in ways that didn't consider the environment, right? And so most of the fights we see right now are about realigning that old water use with our modern kind of sense of what we want to protect in this state and this modern, so this kind of social values and in line with modern law, right? So most of the fights kind of fit within that general idea of conflict and trying to update old water uses. Okay. That's our restrictions on water use. Reclamations, only a couple of slides. Created in 1902, independent bureau created within the Department of Interior. Its whole purpose was to go out and build dams to reclaim the desert in the West for irrigation use. The dams are built for certain authorized purposes. The costs of construction are allocated among those authorized purposes. And this is important because there's a savings clause in Reclamation's act that creates it, the 1902 Act, that says reclamation has to follow state water law. So federal actions normally trump state actions, right? Federal law normally trumps state law, but not in this case where reclamation has to follow state water law. The standard used by the court is that this comes from the US Supreme Court. A state may impose any condition on the control, appropriation, use, or distribution of water through a federal reclamation project that is not inconsistent with clear congressional directives respecting the project. So because these projects have pretty broad purposes for which they're authorized, it gives the state a whole lot of latitude in regulating their water rights and in regulating how the way these projects work on the landscape. We'll skip over the Central Valley Project Improvement Act. It exists. There's a big fight over whether it's been completed or not that I think is getting teed by this administration, and that would take away a bunch of mitigation funds if that did happen, somewhere around $13 to $15 million. So keep an eye on that space, but kind of beyond what we have a chance to talk about. And there's a lot of other federal programs that have impacts on what goes on in the Delta, and we are also going to push through there in hopes of kind of pulling these things together. Okay, so let's talk about how this fits together. We have this water right decision 1641. It was wrapped up in 2000, kind of following on to the 1994 Bay Delta Accords, which led to the Bay Delta Water Quality Control Plan of 1995. So that's under Port Cologne, right? That's a water quality plan from the State Water Board. And then we have this water right decision implementing it. So uses, objectives, implementation done via 1641. And this creates flow, salinity, pumping and operational protections, delta smell and striped bass, which we don't see show up so much anymore for protection. And then ag salinity levels that were set based in part on that Delta Protection Act, right? That area of origin protection for the ag and municipal industrial uses in the Delta. So that happens in 2000. And then we get a whole bunch of other things that come in after that. Right? So we get a bunch of species that are being listed at the state and federal level, and we get BIOPs coming in for those species. We get the CVPIA coming in. We get a whole bunch of kind of adaptive plans on these different rivers, like what we saw on the San Joaquin River with the pulse flows. And we get the Delta Reform Act built in. Let's think just for a second about these ESA issues here. So we have that coordinated operating agreement. That's what was being considered in all those BIOPs. Again, that's section 7 says we have to do BIOPs for federal actions. The state and the federal government agree about how they're going to run the state and federal water projects and the coordinated operating agreements. So you have to do a BIOP every time you revise that agreement. Right, so we saw new consultations for a new coordinated operating agreement that started in 2016. Those BIOPs were issued in 2019. There were no jeopardy opinions. Remember, to bring in the God Squad, you got to have a jeopardy opinion. So if you even wanted to get the God Squad, you'd have to redo these consultations, which I'm going out of order, but which wrapped up again in 2024. You'd have to redo them and get to a jeopardy determination. That takes a long time. It takes even longer if you fired a bunch of your people. So it's not clear to me how that ultimately plays out. Okay, so 2019, new BIOPs are issued. We get a state lawsuit right away. Historically, the state went along with what the federal government did for endangered species. They said, well, if you're complying with the Federal Endangered Species Act, we'll say you're in compliance with the state too. 2019 BIOPs dropped and the state was like, nope, we are not going to follow those. We're going to do our own under CISA. And in fact, we're going to sue because we think that these BIOPs don't comply with what's required under the Federal Endangered Species Act. And that would put more obligation on the state and actually take away water that the state would otherwise be able to use if it had to do more to comply itself. So no jeopardy opinions come out, lawsuits get filed, state environmental groups, court issues a preliminary injunction, and then bam, we've got a new administration that comes in and it says, yeah, these are kind of suspect. We're going to redo these. And so we see those get redone. It takes about 4 years to redo those BIOPs. So we get new BIOPs out in 2024, Fish and Wildlife Service in November, NIMS in December. We've had some species that have been added since then that make things more complicated. But these put a bunch of regulations, a bunch of requirements on delta flows that lay on top of those Port Cologne requirements that we've already talked about. And because the state did its own permitting process, we have CISA requirements at the same time, right? So if we look through, and I stole these slides from the DWR presentation to the Independent Science Board because they're really great. So now it's credited. I guess it's not theft. I don't teach intellectual property. Who knows? So we've got these Bay Delta standards that come from Port Cologne, right? And they're implemented via that water right decision 1641. So state water quality law that controls a ton of what goes on in the Delta. On top of that, this is deliberately too small to see. It's the point is there's a whole lot of requirements that come out of those 2019 BIOPs. I don't have a nice chart for the 2024 BIOPs. I don't think I'm even going to make one because, like, why bother? Consultation will probably start again soon on those. And then we have these state requirements under the State Endangered Species Act laid on top of that, right? So you've got a ton of different mandates telling people who manage the water in the Delta what they have to do, what standards they have to meet. I feel so bad for whoever has to actually like put this together and figure out what flows in what place at what time, because it is so complicated. The best breakdown I've seen of like what's actually controlling at different parts comes from a PPIC report. And it's not even the main report, it's a technical appendix to the report. And they tried to parse out what's actually mandating flows of water through the Delta at any given point. So they looked at 2017, a really big water year, and looked at the Delta outflow and then broke that Delta outflow out to system outflow. That's what's required to maintain the salinity levels, ecosystem outflow, and uncaptured outflow. And so you can see in You can see in a big water year that we've got a lot of environmental water, we've got a lot of salinity water, and we've got a lot of uncaptured outflow. But in a dry year, that delta outflow is almost all system outflow. It's almost all water that's being released in order to meet the salinity requirements in the delta. That's not Federal Endangered Species Act. That's not federal law. That's state water quality law, state water right law that controls that. And this is why even when we saw the big push and got new regulations, new BIOPs written in 2019, there wasn't a massive change in how the delta was operated, right? Because state law and more than that, hydrology is what ultimately controlled how much flow is there. So the amount that you can really flex in this system is surprisingly small, given not the environmental constraints, but given the salinity constraints that end up controlling a whole lot of this decision making. All right. You probably all know the Bay Delta Water Quality Control Plan is getting updated. We have the uses already established for kind of the southern end, and we have objectives derived from those. And the board is currently working on the implementing regulations, so that third level. And we've been hung up on that for a while. Dealing with the voluntary agreements and the healthy rivers and landscapes piece. Not clear where that's going to go except to court. That part is clear, but not clear kind of how it's going to get resolved politically, how it's going to play out. As we're doing that, we're working also on the Delta tributaries and the northern sides of the Sacramento River, establishing kind of all the same pieces. And that's like in play right now. There were hearings last month and will be hearings coming up again soon on that piece. So that's it. That's kind of how everything comes together. I've got a whole list of emerging issues too. And I think I'm out of time now. I'm happy to take questions if you want for a couple minutes or not. I don't want to throw off your schedule with like your first speaker. I get paid by the minute though, so.
$0 a minute. We are technically time for a break, but maybe just 1 or 2 questions since we have you here.
Sure.
Does anyone have a really burning, hot, controversial topic?
I'm not your lawyer. This is not legal advice. This is academic advice.
Hi, Shoko Shinbrat from the Delta Stewardship Council. I'm interested in hearing about what you see as the biggest emerging issues.
Yeah, clearly the Trump administration. And I wrote about this a little bit on the California Water blog. I'm interim director at the UC Davis Center for Watershed Sciences, soon to be permanent director, no longer interim. So that's exciting, but I got distracted. We have a water blog. I wrote about what I thought would happen with the Trump administration that came out kind of right before inauguration. And my prediction was a whole lot of hand waving and anger and rhetoric, and not a whole lot of change in actual flows across the Delta and through the Delta. I don't know, I'm gonna stand by that for now. I think the pieces, so there's been these executive orders that came down, right? And the president has said, we need to look at this is important, we should do these things. Those executive orders don't have the force of law. They don't change the underlying authorities. They don't change what the laws mean. They don't change the requirements of the Endangered Species Act. Right, so it seems like we're headed towards maybe, if I'm going to predict stuff now, a God Squad convening, but you can only convene the God Squad after you get that jeopardy finding, and that's going to take time to do. That God Squad will make a decision, but there are standards that they have to apply. They have to have a hearing on the record to do that. That takes time to do, and then they're going to get sued for whatever decision they make, and that's probably going to tie things up in court for a while. And I don't know how you do this when you're shedding 10% of an already like desperately overworked federal workforce, right? Like you do this, you cut it off. I don't know if the goal is to break government and break it quickly so that then you have some time to maybe rebuild or maybe contract a lot of these things out. I don't know. It's not clear to me like what you're going to do, but if you're shedding people from reclamation, you're shedding the scientists you need at Fish and Wildlife, you're shedding the people you need at NIMS. I mean, the Trump administration did a lot of stuff on the environmental side last time, and they almost always lost in court. They lost like 85% of the time. And that was with really good people working hard. And if you get rid of your good people, and you disincentivize those people who are left to do stuff, I don't know why you'd have a better outcome, a better percentage. So I think there's cause for hope, but it doesn't just happen, right? It takes the state standing up, takes the state meeting its own independent obligations. This is me speaking, right? Not the state meeting its own independent obligations in order to rein in what the federal government would otherwise do. So I'm worried about it. I think it's the single biggest issue. I think there are reasons for hope. I think even the dams that got open, the Corps of Engineers dams where water was spilled, those are dams where there's not an Endangered Species Act hook. Right, there's are dams where the only people you're hurting are the farmers downstream and the cities downstream. So there's not an easy way to file a lawsuit. And we haven't seen an environmental lawsuit over those. Right, so maybe that's where it's kind of like a steam release valve. But that's a very different setting than the Delta, where anything you do has a really big state hook to it. And frankly, I think we get some of the other Western states to back us on that, because nobody wants the federal government telling them what to do with their water. That savings clause that's in Reclamation's Origin Act, there's a similar one in the Clean Water Act. All of those were really insisted on by the Western states before Western state senators would vote for those acts, because they want control of their water resources, just like we want control of our water resources here. But it takes an aggressive state that's willing to do that. Yeah.
I think that's the perfect place to end on. Thank you so much. All right, thank you so much. Sorry to mispronounce your name, that's very embarrassing. We're gonna go to a break and just a 14-minute break. We'll be back at 10, wait, that's the wrong slide. Back at 10:15.
Goes.
Yes, it goes. It goes, it goes.
How are you? Good to see you. I know. I'll talk to you again. The trip we're back in. I guess they. I go on board. So early.
Is it?
Early. It's 10:11, but it's early in the day. I think.
Oh, yeah. All right, we're gonna go ahead and get started as people trickle in. Our first contributed talk session of the day. We've got Claudia Macfarlane to introduce our session on redesigning monitoring.
Good morning. I'm Claudia Macfarlane with the U.S. Fish and Wildlife at the Lodi Field Office. Today for session 2, let's shake things up, redesigning monitoring. Our first presenter is Aroon Melwani. Aroon Melwani is a senior marine scientist and managing principal at Applied Marine Sciences. Aroon has over 20 years of experience in biological study design, sample collection, and quantitative analysis. Today, he'll be sharing an update on monitoring design work focused on optimizing pelagic fish monitoring in the San Francisco Upper Estuary.
Hey everyone. So today my talk is going to provide you with an update on a redesign project that we've been ongoing for the past 4 years, focused on pelagic fish monitoring studies in the Delta. I want to first take a moment to thank many of my collaborators that are working on the steering committee and the design monitoring team. Particularly, I want to call out Steve Slater. He's been working with me very closely throughout the time of this project. Overview of my talk. I'm going to give you a brief overview of the monitoring studies that we are evaluating as part of this project. They should all be very familiar to you. We produced a design review report a couple of years ago. I'm going to provide an update on the major goals and tasks of that work. And then 3 applications that have come since our design review, looking at standardizing the sample frame, working on optimizing abundance estimates, and spatial balance components. So all the work I'll be talking about is done on the auspice of a monitoring design team that has 2 arms. It has the steering committee that was initiated in late 2020, represented by 6 of these agencies who provided the oversight of the review process. We currently meet quarterly with that group. And then the monitoring design team, which has been meeting every month for the last 4 years, working to evaluate and do an analytical review of these studies and develop opportunities for improvement. All the work we've been doing has been conceived around a monitoring design process. I could do a whole talk on just this slide, but essentially we built a planning phase that involved a charter and subsequent work plans that identify the main information and monitoring objectives that are part of this redesign. We engaged with user groups to talk about the intended analyses, and that's all feeded into the survey design evaluation that we've been doing over the past several years. This is a continuous cycle of learning, using our information as well as the information of others to look for opportunities for improvement. We initially set out these 5 main objectives that are focused on improving the data accuracy, the reporting, the interpretation, and the spatial coverage of these studies that I'll be talking about. And today I'll mostly be hitting the first 3 on this list that talk about the habitats and species that are the focus of the analysis, the ways that we're looking to improve the metrics of uncertainty and fill data gaps. So all of the studies I'll be talking about were initiated many, many years ago, starting as early as 1959 with the Summer Tonet survey, with the regional state water project, and then the main state water project that came on when the Falmouth water trawl began, and then the more recent studies that have been more species specific, the 20-millimeter survey, and then the smelt larval survey. We initially had spring Kodiak trawl as part of that till it was discontinued. As you'll see from this figure, they're all kind of spanning different timeframes. So generally we focused on the post-pod period 2002 and since to kind of focus our analysis on the timeframe when all of these studies were working and we could evaluate these opportunities. We initially conceptualized them as these individual studies. They all have their own separate spatial frameworks, and they all inform annual recruitment, provide information about entrainment risk. But as we started to think about these studies together and wanting to relate them, we knew we had to put them on a consistent footprint. And so that's what we've been looking to do, to align these studies so that we could look at them and evaluate them together to look at those gaps, redundancies, and look at them on the same sample frame. So our initial review identified many of the things that we've seen in other IEP reviews, that there's a lot of similarities amongst these surveys. They collect tow information, station-specific information. You can extrapolate that to CPUEs, and then you can calculate an index of relative abundance, but historically they didn't have uncertainty associated with that. So that was a lot of the focus of our work, was how do we bring that into our analysis and help with the interpretation of these data. But at the start of this work, we looked at the individual surveys and the calculations that they are using to extrapolate that catch data, and they were all different. They used different spatial scales, they used different expansion factors, and so the reporting was by design. And so we looked at ways to integrate that information so they could still be by design, but provide a consistent framework. And so our main findings, initially, we produced a report in 2022 that talked about these 4 elements that we've been working on since that time. A very consistent sample frame would be helpful. All of these surveys sample in open pelagic water deeper than 6 feet, which is generally where the vessels can go to. So we needed a sample frame that was explicit to that. We also needed to answer questions about random sampling. That was one of the main questions that was brought by the steering committee to us. And so we needed to design a study to look specifically at that, and I'll show some results. And then we really felt like we could incorporate design-based abundance estimates. So incorporating uncertainty using the actual specific survey designs, but using it in a very standardized framework could be applied. And there are other issues around connecting those surveys, like life history and gear efficiency, which could be worked on later. And then we thought that there are opportunities to improve spatial balance both within and amongst these surveys. So we've been looking to make progress in that area. So the first thing we did was really build this spatial framework. It was initially conceptualized based off of the building blocks of the EDSM program, the Enhanced Delta Smelt Monitoring Program, that provided these 35 subregions. They're the very light yellow colors you'll see on this figure. And then we did cluster analysis to look at how do we aggregate these. And we came up 2 spatial scales of strata, which are the colors that you're seeing here, and then the regions, which are the dark yellow boundaries around them. So there were 7 regions and 12 subregions that we refer to as strata. And then it incorporates the foundational aspects of our redesign, includes the depths. It's explicit about the depths that these surveys sample in. It is explicit about the volumes that are associated with that. And then we constrained all of our calculations to where these surveys actually go, which is generally in 6 feet or deeper water. We included volume calculations, building off the latest bathymetry data, working that through R. We're able to calculate explicit volumes, surface areas, and then and what we're using for our expansion factors is the 0 to 10 meter water column bin that is associated with these volumes. And we excluded any of the waters that were too shallow for these waters to be accessible for surveying, and so they're not included in our expansions. We produced a design team report in 2023 that talks about all of these foundational aspects, the calculations, and the volumes that we generated at that time. We're updating that right now with some changes that we've made to the polygons, as well as extending our sample frame into San Francisco Bay as we're looking at evaluations with the San Francisco Bay study. So there's a table in this technical report that we've produced, and we'll be updating it later this year, that looks like this. So it gives us our surface areas, it gives us our 0 to 10 meter water bins, and it gives us our total volume. These are what we're using for looking at design-based abundance estimates and looking at spatial balance. And so this was the key point to our redesign to move forward. One of the ways that we used these results was in a special study. So I mentioned that we wanted to address the question of any bias in the fixed station monitoring design. If we add stations, want to modify the design, should that be done in a randomized way or continuing with the historical geographically fixed station monitoring? So a fall mid-water trough special study was designed in 21 and 2022 to look at this question. In 2021, it was a very small subset of strata that was sampled from San Francisco, from San Pablo Bay up into Sassoon Bay, Sassoon Marsh. And in 2022, this was expanded all the way up into the deep water ship channel in Cache Slough. And in 22 specifically, it was sampled in a way that allowed more data comparability, a same day, same bay analysis. So this is an example of some of this output. We're currently writing it up in a technical note that's being led by Tim Malnick. What we found was generally a lot of similarity. So you can see on the left side here for San Pablo Bay and Upper River, there's a lot of similarity in the fixed station and the random stations. We saw a very similar pattern in the confluence. In some of the other regions, it was up and down. Some months we saw the random stations to be higher, and sometimes the fixed stations were higher. So there was no consistent bias. We weren't always seeing one type of sample site being higher or lower than the other. We saw a similar pattern when we threw all the data together. This is an example of an NMDS from the 2022 data. So you can see that the over ellipses are overlapping, suggesting that these two samples type sites are similar. We did see some subtle differences. So you'll see on the left side of the plot are a lot of the triangles. Those are the Sacramento deep water ship channel sites. They seem to be driven more by turbidity, whereas the other sites are kind of mixed all in together, which is what we would have expected to see if there's really no difference amongst these sample sites. So our conclusions were that there really doesn't appear to be any bias in the way that the historic sites are being sampled. They seem rather similar to the random sites. So the next application that we did was, once we take these volumes and this design that we have, is how do we standardize the design-based abundance estimates? So this is how we extrapolate those catch data to say how many fish exist with variability in the subregions of the Delta. And so the point of this plot is to show that these surveys sample a very wide variety of species. Summertona has over 65 fish taxa, Some of these are going to be appropriate for design-based abundance estimates, and some will not be because they are just too uncertain to be able to generate good estimates. And so the way that we approached this was building these distribution maps. So these talk about both the life history of the different species, as well as the species that are caught in sufficient numbers to be able to generate these abundance estimates. So these are the species we've generally focused on in these pelagic fish surveys to compare the effort and what uncertainty there is in certain species. The approach that we're taking to have a consistent index methodology is built off the work of Fish and Wildlife Service, particularly Leo Polanski's paper from 2019, that presents the design-based abundance estimates. And so we're basically adapting this approach using our extrapolated volumes, using the fish catch data from the CDFW studies to build in these estimates that are going to be comparable amongst surveys eventually. And so we've built this shiny app that allows us to use any of these surveys. And I've, in dark orange here, highlighted the base study, as that's work that we're doing right now to incorporate the base study once we finalize our sample volumes for them. We're going to be able to generate estimates at the sub-regional scale, stratum scale, region, or delta scale associated with variability. A key component of it will be to compare how these historical indices compare to the design-based abundance approach that we hope to use in the future. And this plot shows two examples of it for the longfin smelt and Asia striped bass that generally show a lot of correlation. We would expect some differences because we're using different expansion factors. So the fact that we generally see the same peaks and valleys in the data set is good, but there will be some differences as we use these different factors. So then taking to the third application has been working on bringing together those design-based abundance estimates with our standardized sample frame to look at opportunities for improving the proportional sampling. Are the areas that are being sampled, are they balanced spatially? And so we have a lot of inputs to that. Some of them I just talked about, the sample frame, the volumes that we've calculated. We have the catch distributions of those specific species that we know that we can work on. And we're bringing those into several analyses that are the activities around our sample frame. And so we're doing analyses to look at spatial balance, and we've got several outputs, and I'll show you the quick examples of those. One of those has been to expand the SLS and 20 millimeter into San Francisco Bay, into San Pablo Bay, excuse me. So this was to fill a data gap in where the longfin smelt tend to occur downstream in wetter years. And so these stations were balanced with the effort that's currently conducted in Sassoon and Honker Bay, which tends to be the center of the longfin smelt distribution more in dry conditions. So now we've expanded it down further downstream. We also filled data gaps in the fall midwater trawl in 3 specific areas. It wasn't possible to generate good uncertainty estimates because there were too few stations. So again, we're here filling data gaps. And the way that we filled those data gaps was leveraging a lot of existing historic stations. So here you're seeing on the right, Sassoon Marsh, where 3 stations were filled in using historic 20 millimeter and fall midwater trawl stations. So that's going to help with the spatial balance and calculating uncertainty for those regions. And then the work we're doing now is to incorporate base study. So again, this is specifically looking at spatial overlap between the fall midwater trawl in San Pablo Bay and going further upstream. But we also wanted to standardize the way that the data gets interpreted into San Francisco Bay as well. And so that's the work we've been doing most recently. So this is going to help inform both standardized reporting for base study that's going to align with these other surveys, and also allow us to compare spatially the effort that's being conducted by the 2 types of surveys. And so the progress we've made has been to be very explicit in our sample frame about the habitats and then the species that are going to get reported through that analysis. We've updated the work that has been done by Leo Polanski and others to develop metrics of uncertainty and fill several data gaps in surveys and times that will now allow us to look more specifically at the other 2 components, which are the statistical resolution and the overlapping sampling that we're working on right now. And so I'll just take you to the end. We've got several ongoing goals. I've talked about several of these. Largely, we're looking to integrate base study with our existing sample frame to look at overlapping sampling and how to improve the spatial balance. And we have several technical notes that are in progress that we're hoping to publish on this work. And with that, I can take questions.
Would you mind speaking?
Oh, sure, yeah. Curious if you're looking at discharge and velocities as part of your sampling scheme. There's been evidence to show, different tidal variability can produce higher numbers of catch in certain regions and stuff like that.
Yes, we are. Not specifically flow, but we are incorporating tidal range into the way that we calculate the volumes. So we're not just taking the mean low water, but we're actually expanding to account for the mean tidal range. And the current analysis has used the same way of expanding tidal range as the EDSM program has used, which is based off of the range at Antioch, which was at the time calculated to be 1.25 meters. So we've increased the volume around the bay to incorporate that range. We're currently looking at how appropriate that number is for some of the more downstream areas, particularly as we're thinking about the bay study. So we might need to have a bigger range because of that range the range in tide down in the South Bay can be much greater. So we're incorporating that in that way.
Next, we have Adam Nanninga with the U.S. Fish and Wildlife Service.
All right. Thank you, everybody. Thank you, Claudia. Like she said, my name's Adam Nanninga. A little bit about me. I have 13 years with the U.S. Fish and Wildlife Service, a little over 8 with the Delta Juvenile Fish Monitoring Program down in Lodi. And for the last 2.5 years, I've been the program lead. This talk, just as a heads up, it is in no way a commercial for U.S. Fish and Wildlife Service or the Delta Juvenile Fish Monitoring Program. It is really just a focus on the value of an IEP review that we went through a little over a decade ago and how we've been able to implement some relatively small changes over time that lead to pretty significant increases in the efficiency and usefulness of the data that we've collected. So a little bit about the Delta Juvenile Fish Monitoring Program. Our main objective is to document the abundance and distribution of juvenile Chinook salmon in the Delta, with a special emphasis on federally endangered winter run and federally threatened spring run. We also need to comprehensively monitor year-round for all the races of juvenile salmon that are present. We intensively monitor in the winter for potential use in water operations, and I'll go into that a little bit more in the next slide. We need to document the abundance and distribution of federally threatened steelhead. We want to look at the abundance of non-salmonid species, and then we need to identify factors influencing survival of these species in the Delta. So the Delta Juvenile Fish Monitoring Program, it's been in operation since 1976. While the monitoring design has changed slightly over the years, this is the basic summary of what we've been up to the most recently and for the longest amount of time. So this map shows that we have trawling 3 days a week for mid-channel monitoring at the entry points of the Delta. So there's 2 circles on the Sacramento and the San Joaquin, and then 1 spot of trawling at the Chips Island, which is the exit point of the Delta. We also have littoral sampling using beach seines in 6 distinct regions throughout the Delta and 1 seine run in the Bay area. Those are all the other spots there. And we also have a 7th very important seasonal seine run that's in the Sacramento region. And that is specifically designed to detect winter run juveniles coming down through the Sacramento region. And it could potentially lead to water operators making decisions related to the operations of the cross channel in Walnut Grove, just slightly downstream of Sacramento. So a little over a decade ago, we went through a IEP scientific advisory group review. And they had 10 main objectives of the review. Basically, does DGFMP have clear objectives and goals? Is the sampling design and data collection effectively answering our study questions? Do we have adequate sampling, both spatially and temporally? Do we sufficiently monitor for ESA compliance and trend assessments? Is the information we're collecting useful for other species and for total ecosystem understanding? What are our limitations in our data? Are there different formats that our data uses over different time periods? Is our data analysis accurate? And is the interpretation of that data accurate? What's our frequency of reports? And what is our actual measure of success? And are there general recommendations for program improvement? And so while I can't go over, I don't have enough time to go over all of the results of the review and how we've implemented change. There are 6 major portions that we did take a look at really closely and implement. So the first one is that we need abundance, not just abundance index. And so abundance index is basically our amount of catch per the amount of water that we're sampling. So we have both those measures and we calculate an index, which has been very useful over time for looking at trends. However, what we actually need to know is how many of these listed species are at those sites at those times. So we need actual abundance. Seines, they only sample limited habitats. So you can't seine in vegetated areas or places where logs have washed up, things like that. And so the usefulness of seine data can be fairly limited. As we know, it's well documented that the Delta is becoming overrun with aquatic invasive, vegetation. Our data is somewhat lacking. The QA/QC process was outdated at the time. We weren't taking full advantage of technology available. We need very accurate net measurements because both abundance calculations and catch per unit effort are going to rely heavily on actually how much water we are sampling. We need to look into new technologies to advance. And then we need to also look at fish community, ecology, and life cycle. So now I'm going to go through one by one and talk about how we've addressed these concerns. So calculating actual abundance at our trawl sites. Brian Mathias took this up. Originally, Pat Brandes was in charge of this, but Brian Mathias is with an offshoot of DGFMP called the Juvenile Salmon Survival Studies. He leads this effort and he worked with Russ Perry with USGS and Brian Piper of Fish Metrics. This is looking at the Delta entry at the Sacramento and exit point at Chips Island, these 2 trawls. And basically the way he handled this was we need to increase our frequency of sampling in conjunction with acoustically tagged released fish released at the same time as hatchery fish. And so when we're trying to look at actual abundance, we need to measure efficiency of capture. And that's exactly what Brian is able to do with this information. So he presented on this last year, but yeah, anyway, for seines, we are also looking at calculating abundance. And this is Brian Mathias, myself, and Jennifer Witt. Jennifer has a lightning talk in the next session that is specifically about the process of increasing the number of seine sites and then randomly picking each week to also help get us abundance estimates. And then we also, for the last couple of years, have been measuring beach seine efficiency. And so this is a really cool project where we actually go out to our beach seine sites. We hang a block net up on poles with a quick release system. We will wait an hour with that net hanging above the water, quick release it, and so it drops and catches any fish inside that have wandered in. And then we will seine 6 times inside the block net, keeping those fish separated in separate tubs, working them up separately. And then at the end, pulling in that block net really carefully to capture any remaining fish inside the block net. And so with this data, we can actually look at how efficient we are with each of those seine hauls. And then also these same sites will be visited at other times and pulled 6 times with a seine net without the block net there. So we can compare those values, not just inside the block net, but without the block net there. I'll mention too that with both the trawl abundance estimates and the seines, one thing that we've started doing a lot more in the last 5 or 6 years is collecting way more DNA samples. We are aware that using the length at date criteria that we've used since the early 1990s is not really great at defining the different races. And so that's another thing that we've really increased. So to address the issue with seines in general, only being able to sample very limited habitats, a few years ago, we started boat electrofishing. Currently, Jacob Stagg in our office runs this project. Originally, it was Catherine Johnston and Ryan McKenzie. Boat electrofishing is much better at sampling all habitats in the Delta. There is some limitations as far as the conductivity that it can sample in. It also is somewhat limited in that it's an advanced technology, but it's prone to breakdown too. And so there are some limitations, but it is still a experimental boat electrofishing survey that is doing really well at capturing fish in all habitat types. It also is able to do truly random. We are able to use truly random site selection. There is also a project going on to compare beach seine efficiency and electrofishing, where basically whenever we do beach seine efficiency with the block net or without the block net, the electrofishing study will either have gone the day before or the day after and sampled the same site. And Autumn Fisher actually has a poster on this very topic of how we're doing the comparison. Ignore the number though, that's apparently, they've moved posters around. So it's not 13A, but it is in methodology assessment and improvement. Our QA/QC process was somewhat outdated at the time of the review. We were doing what was called line by line. So basically all of our data sheets would be entered into an Access database. The data would be printed out, and then a technician would go line by line, looking at the printout and comparing what's on the data sheet to make sure that they match. Many years ago, Andrew Goodman came up with the idea that we should be doing the double entry system, and John Spiegel was able to implement this with help from Morgan Gilbert and Ryan McKenzie, who wrote the code on this. But basically now what we have are 2 databases where a first entry and a second entry database that is compared using our code. And so if there are entry errors, somebody can go back and make the corrections. We also have been sending our data sheets back to the office in real time, so taking pictures and sending them back in. What that does is avoid it by providing the data sheets for people in the office to enter as quickly as possible. We're making the best use of available office time. Many years ago also, we started posting to the Environmental Data Initiative. This was initially by Brian Maharja. And most all IEP monitoring is doing this now, where it's posted to the Environmental Data Initiative, where it's available to anybody with internet access. Previously, before that, it had to be requested. So somebody would have to actually ask for a block of our data and they would receive it. Measuring nets during a trawl. This study is led by Jenny Wiggins, Ryan Cook, Erica Holcomb, and Laura Mitchell. Like I said, volume sampled is absolutely critical for our abundance estimates. So the way we measure volume is basically using a flow meter with an adjustment factor and the dimensions of the mouth of the net. And so EDSM, Enhanced Delta Smelt Monitoring, has done some preliminary tests, and right now we are actually working on, we're getting some more modern equipment to actually look at these while under tow. Coded wire tags are nearly microscopic pieces of metal that have 6-digit numbers imprinted on them, and they are unique to hatchery releases and run type. And so the way that DJFMP uses coded wire tags is that we actually collect every single adipose fin clip juvenile. So historically, 25% of fall run that were from a hatchery had CWTs. All winter run, spring run, and late fall are supposed to have coded wire tags if they're from a hatchery. Up until last season, our process for processing these coded wire tags was staring through this tiny little microscope and also extracting the coded wire tag from the fish, putting on a little sticky tape and filing it away with all the information on data sheets, and then having binders and binders of data sheets. Thanks to Brian Hutchinson and Andrew Goodman, this past year, we've experimented with a device that allows it to be pulled up on the computer screen. The technicians can, while both of them are sitting there, verify the number that's on it, enter it directly into the database, and disregard use of the data sheets. So, and just for, so the range of coded wire tags that we actually collect in one salmon season is from around 2,000 all the way up to nearly 8,000, depending on environmental conditions. So this is a significant reduction in the amount of time it takes to process those samples. We're working on this project we've nicknamed Regions and Seasons. This is Jacob Stagg, Nick Veal, and myself. The basic idea is that we are converting our counts and length data to actual biomass. And then we're planning on working with the University of Washington to put this onto SACPass with a Shiny app. And the idea is that we could actually help the Bureau get a better understanding of how adjustments might affect fish community. And Nick Veal has a poster on this topic. Again, it's in the other fishes section, but not 10B. So some of the summary and lessons learned. Many of these projects are ongoing and we only have preliminary results. We do have several publications in progress for beach seine efficiency, electrofishing, and trawl efficiency. In our office anyway, we had to have a mindset of improvement that comes from all levels. So I want to take a moment to thank Jeff McLean, who's our project leader, Lori Smith, who's our deputy project leader, and Eric Huber, who is our program manager, for really pushing us on these improvements and keeping them moving forward. A little bit of advice for people who are interested in these sort of things. We have to maintain SOPs because long-term monitoring is prone to high turnover. And so maintaining SOPs and project records is absolutely critical. And then work with IEP. Make sure they know of changes and improvements that are happening, and always solicit input from the IEP community. With that, do you know how much time we have? 3 minutes. Okay, I'll take questions.
Hi, I'm Inge Werner from the DISB. I was glad to hear that you are actually looking at DNA or looking into the usefulness of eDNA for these monitoring programs. How far along are you, and do you think you can eventually replace the work-intensive part with more of the eDNA analyses?
So I'm sorry if I misspoke, but we're not actually looking at eDNA. When I talk about DNA, I'm talking about actual fin clips off of individual fish in order to do race determination. I don't know that we've actually done much investigating on eDNA for our projects. I mean, it's on everybody's mind. I think, I'm not sure of the practicality with our sampling, just because they're so interested in specifically abundance and not, I don't know. I don't know. I can't speak to that, sorry. No, it's okay. Anybody else? Okay, we got 1 minute left. Yes.
Oh, okay. Hi, Louise Conrad from DWR. Are there some outstanding elements of from the review that you still want to look at? And what are the hardest parts of the review recommendations that are hardest to implement?
I think, the hardest part of all this is that even these small changes are really hard on our crew. Potentially, they're working really tirelessly on beach sand efficiency projects and things like that. It's not actually that easy to take those DNA samples either, especially when we're getting a lot of salmon. And so these small changes can have real impacts on our crew. And I'd like to just take a moment to thank our crew too for all their work that they do. But yeah, I can't really think of anything in the review that is within our purview that we need to work on still. So thank you for that question.
Yeah, thanks. Brittany Davis, TWR. What hit home was how hard these crews are working and how labor intensive a lot of these monitoring efforts can be. And the review's in 2013, and I was thinking about, AI tools that have been developed, and are your groups thinking or considering or trying to develop any AI tools? I get you have to be in the field to collect the samples, but could wire tags be read and inputted by some computer system, or real-time data collection integrated in? Are any of those considerations, being discussed? There's a lot of work in here.
Yeah, that's a
for those
great
field
question.
crews.
We have not started looking at that sort of thing yet. It's something we should definitely look into, where we can improve our efficiency using AI. I'll hit on the DNA thing one more time. I know that UC Davis is working on Sherlock technology too, and that would be a really useful thing for our crews to have to reduce the amount of work taking fin clips. And so I know they're working really hard on that, and that would be amazing if we could have that in the next few seasons. That'd be great. Sorry, I couldn't answer your question more thoroughly, but we have not, but we should look into AI. Yeah. All right, thank you, Claudia.
Next, we have Kristen Arend, who's a biologist in the Modeling and Water Operations Division in the Bay Delta Fish and Wildlife Office.
Morning. We're going to switch and start talking about zooplankton now, and just take a real kind of surface, sort of broad brush look at how some of the longer term pelagic zooplankton monitoring surveys, how that sampling effort is distributed over space and time. So why do we care about zooplankton in the Delta? They really are of both ecological and management importance. They transfer primary production to higher trophic levels, primarily via pelagic pathways, and they serve as important prey for native fishes. So understanding the quantity, quality, and seasonality of their availability is important to conservation and management. And then finally, they have been shown to respond to environmental drivers, some of which are can be manipulated by management actions, such as flow. So there are a number of different surveys that monitor zooplankton in the Delta and the Bay Delta for different reasons. A couple of the longer ones include the Environmental Monitoring Program, the 20mm survey, Summer Toadnet survey, and the Fall Midwater Trawl. And these all focus more on pelagic habitat. And if we look at these, we can see that they really vary in terms of how long they last throughout a given year. So what their sampling time frame is, as well as the number of samples they're collecting during that when they're out conducting the surveys. So this just shows basically the number of plankton samples collected across a time series for each month. And you can see in green the EMP program samples throughout the year, whereas the other 3 surveys are more seasonal, sampling maybe 4 to 5 months each. But those surveys tend to collect a lot more samples when they are out. And they range in sampling from once a month to twice a month, depending on the survey. They also vary spatially in terms of the areas in which they sample. So you can see just this map is just showing where their sample sites are located with the different colors for the different surveys. You can see that in some areas of the Delta, we only have maybe 1 or 2 surveys out. Other areas of the Delta, particularly the lower Sacramento, lower San Joaquin and Confluence area, we have pretty much every survey out there hitting it and collecting samples at some point in time in the year. So really being able to look across the Delta at zooplankton dynamics and look across the year, it would be really advantageous to kind of pull these data sets together, right, and come up with an integrated data set. And so fortunately, that's already been done. A couple of years ago, a synthesized zooplankton data set was developed, and that allows for integrating data across different zooplankton surveys. And that's been used already to look at long-term trends in abundance, as well as the response of zooplankton to environmental drivers such as flow. But what we were interested in doing was actually looking at the data themselves for one integrated set of surveys, and think a little bit more about how that sampling effort is distributed across those surveys collectively, just to kind of think about what we're getting from this information and from these data. So specifically, we've heard that these fish surveys can take a lot of time and effort and elbow grease. So do the zooplankton surveys. They take time and effort, and particularly identification and enumeration of zooplankton can be really, really time consuming. So we specifically wanted to know if sampling could be reduced temporally or spatially without compromising the information provided. And so to do this, we created reduced data sets of the surveys of this integrated survey data set. We reduced those and simulated relative abundance and patterns in abundance. And then we compared those estimates, or those simulated estimates, to estimates coming from the full data set. And so we would expect that if there is some redundancy in sampling effort, that the reduced data set estimates would match full data set estimates pretty well. But if there wasn't much redundancy, then we'd see some pretty different estimates coming from those different data sets. So we anticipated that we'd actually find pretty low redundancy across these surveys. And that's just because of what I already talked about. Survey overlap is really just concentrated in a subset of subregions in the Delta, and sampling effort varies seasonally, with the highest effort being in the spring and summer. And I'm just showing that in this table on the left, where I have for each season which surveys are out sampling for at least a month during that season, and then the relative sample effort. And you can see there are a lot more samples collected in the spring and the summer. So if there were any redundancy, we expected that to be in the spring and summer when we have our highest effort, and also in those subregions where we have a lot more overlap in those surveys. We also expected redundancy to be low because as you can see in this plot here, that's showing the catch per unit effort by season across different taxonomic groups. There are some strong seasonal patterns in community composition, and also just based on kind of the error bars, you can see there's a lot of variability in zooplankton CPUE. So we anticipated we'd need quite a few samples to kind of capture that variability over time. So how did we go about looking at this? We followed the approach that the long-term monitoring element review used a few years ago to evaluate multiple Bay Delta fish surveys. So first, we identified some representative taxa from the zooplankton community, and then we used the full data set to identify the kind of best model or select the best model that would allow for simulations of abundance over time. Then we created reduced data sets and compared the estimates of abundance from those reduced data sets to that full data set to see how well they performed. So again, first we needed to come up with representative taxa. So we used a principal tensor and silhouette analyses to do this. The principal tensor analysis is a type of cluster analysis, and the silhouette analysis measures how similar species are to others in their cluster. So on the right, you see this dendrogram, which is basically just showing the different clusters that we came up with in the different colors. And then we wanted to select representatives, one representative from each of those clusters. And that could largely be based on the relatedness of that taxon to the other taxa in its group. But sometimes we had some that were pretty similar. And so then we chose ones that we knew were important fish prey, or ones that we knew were important from a management or ecological perspective. And that was based on expert opinion from folks mostly in the Zooplankton project work team. So a little pitch for the IEP project work teams because they were very helpful. So we have our representative taxa. The data set that we used, we pulled from Zooper, that integrated data set synthesizer, I guess. And so we used data from 2004 to 2021 just for EMP 20 millimeter summer townet and fall midwater trawl. You can just see from these quick plots of histograms of CPUE, we have a lot of zeros in these data. So they're really zero inflated data. So we used Bayesian log normal hurdle model, and we tested out a couple different configurations of that model. So we included year and season or month and surface salinity or subregion. And then we used leave one out cross validation to select kind of the model that fit that best. So then we needed to create our reduced data sets, and we did this spatially and temporally. So I'll talk about our spatial reductions first. So we divided the delta up into the subregions, the same subregions that Aroon talked about earlier. And so we can pretend that these little pie pieces are each a subregion of the delta. And then we randomly assign stations into 10 groups within each of those subregions. So each of these dots is one of those 10 groups that has these randomly assigned stations. And then we could basically pull the groups, remove groups from each of those subregions to come up with these reduced data sets. So for example, we wanted to reduce the data set by 10%. So we removed one of those groups of 10. And we can do that for each of those groups of 10, which gives us 10 different reduced data sets. We also did the same thing to come up with a 33% reduction, which would give us 3 reduced data sets, and then 67% reduction in data, which also gives us 3 reduced data sets. For our temporal reductions, we split the data up into months and season, and then we removed either 1 month per season or 2 months per season from the data set. And so by removing 1 month per season, we end up with about 33% of the data removed, so comparable to those 33% spatial cuts. If we remove 2 months, it's comparable to those 67% spatial cuts. So then we needed to fit the reduced models to the models that we had, or the reduced data sets to the models that we had selected, and simulate abundance over time. And so what we could do is use that. So here I'm just showing CPUE over the time period, the simulations from the reduced models, and the full data set. The line is just showing the median, and then the 95% credible intervals are shown in the shaded areas. So now we have these different estimates of abundance from the reduced versus the full data sets. And we can also calculate trends in abundance. So basically, how much did abundance change from one year to the next? Did it increase, decrease by how much? And we could compare that as well. So the comparisons that we did is basically, if you think about overlying the reduced model and the full model, you can then basically calculate the percent of the reduced model predicted values that fall within that 95% credible interval of the full model. And so I'm going to summarize this graph on the right because that's basically, you're going to see a lot of these in the results. You're going to get sick of seeing them. But what I have here is the proportional overlap with the full model on the y-axis, and then the proportional reduction in sampling effort on the x-axis. The station or spatial reductions are shown in triangles, and then the month reductions, the temporal reductions are shown in circles. We have the 95% overlap just in that dotted line, just kind of for reference. And then every data point that you see represents a year, but it's like the average across all the reduced data sets for that year with the standard error. So that's what we'll be seeing a lot of. So what did we find? Well, first of all, what were the models that we selected? It broke out into two different models for different sets of the taxa that we selected. So Pseudodioptimus forbici, Acartia, and Tortanus were all best described by a model that included month, year, and salinity, whereas Daphnia, Cynocalanus, and Acartiella sinensis were the best model included subregion instead of salinity. So those were the models that we were using. And then what did we find? So if we can just start by looking at Cynocalanus first and thinking about overlap in the abundance estimates, so CPUE. So again, each of these dots is showing a year. And so the spread in the dots are going to be, variability for each of those years. And the error bars are going to be variability across those different cuts or the different data sets, reduced data sets. So we see right away that the 10% reductions didn't, impact the data set too much. I mean, we're within about 90–95% overlap with the full model, but then that starts to decline with greater reductions of the data set, which isn't surprising. We do see spread with, across years. So some of that, could have to do with what's going on in a given year in terms of the abundance of the particular taxa. And also, I think it indicates that which stations or which months are removed matters as well, just because of those error bars. So we can look at all of the 6 taxa together, and we see that it's pretty similar in terms of the patterns that we're seeing. We do have some cases where we have maybe a little more spread in those 10% cuts, so not all of them performed as well as the others. And then there are some species like for P. forbici, those month cuts really seem to reduce the proportional overlap with the full model compared to the station cuts. But that wasn't always the case, as you can see in other instances, they didn't differ that much, or even sometimes the station cuts were a little bit worse. So I'm not going to ask you to look at all 6 taxa the rest of the time because your eyes will glaze over. So we're just going to focus on Acartia and Cynocalanus because they're pretty representative of what we're seeing. And so again, if you remember, I said that we might expect some of that redundancy to be found in the spring and the summer when we have a lot higher sampling going on or a lot more samples collected. So we wanted to just break this up seasonally, which I'm showing here. So for each season, what that proportional overlap is for each reduction in sampling effort. And we really didn't see, less redundancy in the spring and summer overlap. So, we would expect to see more proportional overlap if that higher kind of effort of sampling in the spring and the summer was really creating redundancy in sampling. And then finally, I'd mentioned looking at trends in abundance estimates as well. And those fell out really similarly to the CPUE estimates. So again, we're seeing that we're not losing a whole lot with our 10% cuts, but once we get to the 33 and 67% cuts, we're at times dipping below 75% and even below 50% in terms of the overlap. And we saw the same patterns with seasonality as well. So our preliminary conclusions, and we have a lot of caveats to this, is that there is some, but not a lot of redundancy in these, these integrated data sets, right? Again, thinking across the surveys that we included. So there could be some potential for reduction and maybe reallocation of effort from those times and places where you have a lot more sampling going on to those times and places where less sampling is going on. So that's something that could be dug into and considered. We haven't looked into that at this point in time. So really all we were looking at here was kind of real surface level across surveys and delta wide, what these combined data sets tell us, and then what their relative effort is. We didn't look at the impact to individual survey data sets that would happen if we started removing sites or reallocating sites. So we haven't gotten into that level at all. This is really only applicable to estimating abundance and trends in abundance, at least our analysis is, but we know that these zooplankton surveys are set up for a number of different objectives. Some of them are really focused on looking at fish diets and prey availability, maybe prey selectivity by fish. Others that we didn't include in this analysis focus on restoration success. So it's important to think that it's, your objectives probably matter in terms of sampling effort and any redundancy that might exist. And then, this is applicable to zooplankton only, but these surveys also collect a number of other parameters that would be impacted by any reduction or reallocation. And also, you could probably look at those parameters similarly. And I think with that, we have the big thank you to all the field and lab crews that spend so much time collecting and counting and identifying all these critters, and the data managers as well. The developers of Zooper, and then we had worked with two different advisory groups. We worked with the Zooplankton Project Work Group, and then we had a broader stakeholder group as well, providing us some advice along the way. And if there's time, I'm happy to take questions.
Can you hear me? In your second model, was there any sort of theory why spatial information like subregion was a better predictor than environmental information, say temperature?
Yeah, so I think, and I think there are probably people in the audience who know each of these species better than I do, but what we were thinking is that it really was, especially looking at Daphnia being one that where subregion worked, that those are just that much more strongly associated with a certain salinity, and we have such a strong salinity gradient across those subregions that the subregion was able to capture that kind of salinity tolerance as well as, just as well as the salinity model would. And the subregion, because there are so many fewer subregions than the data we had for salinity, those models were faster and tended to fit the data a little better. So.
That was really interesting, Kristi. Thanks so much. I was wondering, do you think the data set that you guys built would be useful for looking at community level shifts over time too? And if trends in community, because the zooplankton community has changed so much over the time period that you were looking at. Do you think that the kind of data reduction and all those types of steps that you took for these type of analyses would also be appropriate for looking at community level shifts?
Yeah, I don't know if the approach would be good for looking at the shifts. I think that by looking at these different taxonomic groups, you can understand how any reduction or reallocation might impact your ability to detect those community shifts, because reductions might impact your ability to really understand what's going on with the abundance of one zooplankton better than another. I don't know if that answers your question. All right, I think we're out of time, so thank you.
All right, folks, we are running a little bit long, but we'll just take a quick break and be back at 11:20. So quick stretch, fill up on coffee. Be back soon.
online audience that you can ask questions anytime during the presentation through the Q&A button on Zoom. We'll try and rotate between your online questions and in-person questions. Our next session is new monitoring and expansions. Our first speaker is Vanessa Lowe. She is the field lead for Pacific States Marine Fisheries Commission's White Sturgeon Monitoring Program and a contractor for California Department of Fish and Wildlife. Is that not showing?
Oh.
That's not sliding.
No, it's, yeah, it's like, yeah, that's
Sorry
okay.
about that. I'll show. Thank you.
Hi, everyone. I'm going to talk about a new fishery-independent survey method that CDFW implemented last year. So brief overview. So today I'll talk about the brief biology of white sturgeon and then the history and the monitoring that CDFW has done to date so far. And then I'll talk about our new marker capture pilot study, compare it to some of the previous monitoring that has been done, and then talk about our plans for the 2025 field season. So sturgeon are this group of ancient fishes, and almost all of them are at risk or endangered. In North America, we have 8 species. And then in California, we have 2 species. Both are anadromous. We have green sturgeon, which are generally more marine oriented. They've historically had a lower abundance. And then, as you may have heard earlier in the day, the southern distinct population segment is protected as threatened. And then we have white sturgeon, which are more estuarine and freshwater oriented. They can be found in some of these major rivers. And then in California, they spawn only in the Central Valley in these kind of green boxes. It's questionable whether they spawn further up in some of these tributaries. And then as of last July, they became a candidate species under the California Endangered Species Act. And so white sturgeon exhibit really slow growth, a late age at maturity, and they only spawn periodically. So in this plot on the right, you have age on the X and then length on the Y. And they, in order to become sexually mature, they're probably or roughly like 5.5 feet long. So I'm 5'2", so that's like a fish that's like a little longer than I am tall. And then the orange dashed lines are the previous slot limit, so the size at which anglers were allowed to take these fish. And then they also have infrequent recruitment, and their success appears to be correlated with wet water years. So on this plot, we have year on the X, and then mean daily outflow is in blue during the springtime. And then the gray bars are the year class index, which come from the Bay study, and they're age 0 and age 1 sturgeon catch. So you can see with in the more recent years, with more frequent and longer droughts, we've had fewer years of high recruitment. And so a little bit about the history and the monitoring. So California actually used to support both a commercial and a recreational fishery. And the first sturgeon closure actually occurred in 1901. And then it was periodically opened and closed. And then in 1918, it was closed for a longer period of time. And then in 1954, it reopened for both green and white sturgeon. And so just to kind of summarize, like the main management tool that was being used at the time was just to alter the length at which people could harvest fish. But there weren't that many restrictions on the number of fish that could be taken. And so the population continued to decline. And in 2006, green sturgeon were listed as threatened under the Federal Endangered Species Act. And that triggered a closure of the fishery in 2007, and also an implementation of sturgeon fishing report cards for white sturgeon. And then in 2022, there was a really big harmful algal bloom, and then a subsequent smaller one in 2023, which also triggered a California Endangered Species Act listing petition, which was brought by a number of nonprofits and members of the public. And so just to emphasize how bad the 2022 harmful algal bloom was, there was at least 864 sturgeon carcasses that were found. A majority of them could not be identified to species. And then in 2023, we had much fewer. But just as like a note, this monitoring at the time for the carcasses was very haphazard, and CDFW is just walking shoreline to locate carcasses, and they walked maybe 20% of the shoreline. And as aquatic animals, of course, you expect that many of these bodies probably sank or were otherwise not physically seen. And so in 2024, the Fish and Game Commission unanimously approved that CISA listing petition for white sturgeon. And so that initiated a status review for white sturgeon. And then also in the interest of trying to continue recreational fishing for white sturgeon, CDFW brought an exemption which allowed catch and release, following findings from other watersheds that catch and release fishing for white sturgeon was not too stressful, and was still an appropriate way to allow the public to utilize this resource. And then at the same time, last year, we implemented the set line survey pilot study. And so previous to the set line study, the monitoring that was occurring for white sturgeon was just the sturgeon report card program, which was if you wanted to fish for white sturgeon, you would have to go purchase a separate fishing license, and then you would go fish and you were supposed to mail it back to us. And then the second one was this reward tagging program. And so CDFW went out on boats and trammel netted for sturgeon, and they would tag them with these tags that actually had a monetary value. So if a fisherman went out and caught a sturgeon, they could mail this tag back to CDFW and receive that money. And so this information gave us data on exploitation rate, angler behavior, population estimates, and distribution. And this is the population estimates over time so far that CDFW has collected. The blue triangles are from the Lincoln-Peterson equation. And then you can see in 2007, when they implemented the reward tagging, then they created a Lincoln harvest estimator. And in general, the two, in the years that they overlapped, they did agree with one another, but you can see that the precision in the Lincoln harvest, where it's not as precise, whereas the Lincoln harvest estimator is more precise. In the more recent years, you see that that precision is becoming wider, and that's because fewer people were actually returning tags to us. And so the current 5-year average that CDFW has is from 2017 to 2021. And they estimate there's about 30,000 legal sized fish. So that specifically is the 40 to 60 inches that anglers were allowed to take. And that's a decline from an estimated 150,000 in 1997. And so although this was these like sturgeon report cards and trammel netting was the best available data we had, there were some limitations in that we had low participation rates. So people would not return their tags to us or their cards to us. And then in some cases, even though the reward tags had that monetary incentive, they would still either not return them, or sometimes the data was just incomplete. And so that kind of really made it obvious that a fishery-independent study was important. And that is why we implemented the white sturgeon marker capture pilot study. And so this was modeled very closely from a joint Oregon and Washington Department of Fish and Wildlife survey in the Lower Columbia that they've been doing for over 2 decades. We modified it a bit for our waterways and for our local community. And so one of the big things that we did was we partnered with local sturgeon fishing charter captains, in part because they know the waters very well, and then also because some of them have been fishing for sturgeon for multiple decades and they knew where to find the fish. And so the study was separated into 2 phases. We had the marking period, which was in April and May, and we had 2 boats out on the water. We're out there 4 days a week and setting 30 lines per boat per week. The study area was based on the highest sturgeon report card capture rates. So in the map, we have the colors represent kind of different regions, and then we further split those regions into 9 kilometer square grid units, as I'll call them. And then in the recapture period, this was from August to September, we were able to add a 3rd boat. And similar schedule, 4 days a week, 24 to 38 set lines. But we did try to sample randomly. So we used grit sampling to determine a spatially balanced sampling schedule, but we did weight each of the units depending on higher density zones, which was based off of the proportion of catch from the sturgeon report cards. So kind of like what we expect the distribution of sturgeon to be. For this year, we will be modifying the weighting based off of our 2024 field season. So hopefully it'll become more in alignment with the actual distribution of sturgeon. And so the gear that we use, we have these 600-foot long lines with anchors on either end. And then we have 40 hooks that are spaced out evenly on the line. And those hooks are in multiple sizes. We've got 3 different sizes. We made our own pickled squid bait, and then we used a little bit of lamprey as well, based off of our boat captains insisting that lamprey was way more efficient than squid. And then in total, over the 4 months that we were out there, we deployed 40,000 hooks. So when we caught a sturgeon, we would collect various morphometrics like length, girth, weight, and pit tag number if they had one. If they didn't have one, we gave them one. And then we noted their condition and location. And then for environmental data, we collected things like water temperature, DO, salinity, connectivity, tidal state, turbidity, and depth. We did get some bycatch. And so for bycatch, we identified them down to species, count, total length, and condition. We mostly got sharks and rays, as you'll see in this picture. There'll be more pictures later. And then despite all the happy smiling pictures, we did have some problems. There was ropes and props, and that is a tree trunk that snagged one of our lines. And then we had some fish that were enormous that we could not remove from the water at all. So our catch by sampling unit, we were successful in catching sturgeon in almost every single grid we sampled, with a majority of them occurring in this central region, which is Sassoon, Grizzly, and Honker Bays. In total, we saw 1,451 sturgeon. Some of them jumped off the line or we weren't able to tag them. So of those, we were able to tag 1,364. And then on this histogram on the right, we have fork length, and the dashed lines are the old slot size, and then the gray bars are the kind of range at which sturgeon becomes sexually mature. And just as like a caveat, like the old slot size, there's a couple reasons probably. We don't know for sure why it looks like that portion of the population is less, but it could be due to that size class was pretty heavily affected by harmful algal blooms. And then also it was the slot size for take for 9 years. And then on top of that, there is like a pretty good chance that, or not a good chance, but that slot size group is likely part of a 2011 cohort, which was not very successful, whereas the fish that are about to grow into the slot size limit are from a relatively successful 2017 cohort. So one of the things that we were able to look at, because we collected weight on some of these fish, but not some of the larger ones because they're really big and heavy. So we looked at kind of like the length weight relationship specific to our population. And so this will help us in the future be able to kind of, when we have this relationship, we'll know how heavy fish are when we catch them. And then we also overlaid it with the Blackburn age data. And then for reference, again, you just have the sexually mature gray bars right there. And then for fish that we actually recaptured, this is just a representation of a couple of the shortest and longest traveled recaptures that we got. As a reminder, the colors represent the regions, and then we have the separated grids for each of the regions. If it's a circle, it was captured in our marking period, and if it's a triangle, we captured it in our recapture period. Last year, we had 1 month in between field seasons, but next year, or this year, we plan to have 2 months in between our field seasons. And then for reference, the minimum number of days between capture was 29, and the maximum number of days between capture was 129. And this confirmed to us that our fish were appropriately redistributing in the system, which is an important assumption of a Lincoln-Peterson estimate. And you can see, so we have kind of like this yellow fish in the middle, barely moved, and then the fuchsia, lime green, and dark blue fish moved quite far. And then we have one fish that was actually captured 3 times. So the red one was captured twice in the marking period and then once in the recapture period. And then here's just a little bit of the bycatch we got. So mostly we got sharks. We got leopard sharks and smoothhounds. One of the cooler ones was we got some guitar fish that's in the bottom right. But importantly, we had very little mortality of bycatch. We had relatively low striped bass catch. We did not catch any green sturgeon, nor did we catch any salmon. And for comparison, the trammel netting that was the previous method of sampling white sturgeon, their greatest bycatch was salmon. And then here's just a little spatial map of the bycatch we caught. And since you saw that it was almost all sharks, most of those sharks were caught in San Pablo Bay, and then striped bass, catfish, and anything else was mostly caught in the Sassoon and lower Sac areas. And so to compare it to the trammel net, in terms of sturgeon caught by survey method, this is in sturgeon per boat per day. And we found that our set line method was quite a bit more efficient than the previous recent years of trammel netting. And so for 2025, some of the things that we are about to implement, we're going to include sonar surveys for white sturgeon, and this is going to be based off of NOAA's Southwest Fisheries Science Center green sturgeon sonar surveys that they do annually, I believe. This is a side scan sonar image of an aggregation of green sturgeon. And so each of those little dark shapes is a sturgeon. And it's like a 5 or 6 foot fish. And we'll be doing this in the regions that are known white sturgeon spawning areas. Additionally, we're going to add a 3rd boat to our marking phase, which is that first 2-month period. The reason being that the greater number of initial marks increases the accuracy of the estimate. And then additionally, we will be acoustic tagging and sexing a subset of these fish, just visually. So we're going to use it when we make that incision to insert the tag, we're going to put an otoscope in there and actually look at the gonads and image them. In some cases, we'll even take a biopsy. And with that, I would like to acknowledge a very long list of people that helped on this project. We have mostly CDFW and PSMSC team on the left, and then these are our contracted captains and deckhands on the right. And with that, I will take questions.
Actually, we're pretty much out of time.
Yeah,
Sorry.
okay.
Ask Vanessa in person. Our next speaker is Craig Stewart. He's an environmental scientist with the California Department of Water Resources.
Morning, everyone. Almost afternoon. My name is Craig Stewart, and I'm an environmental scientist with the Department of Water Resources Discrete Environmental Monitoring Program. And thank you for tuning in. My title, or the talk, is titled Spatially Explicit Water Quality Data Collected on Board the Sentinel. So the environmental monitoring program is a joint effort between the Department of Water Resources and the United States Bureau of Reclamation. And its primary goal is to characterize the aquatic environment of the Sacramento, San Joaquin Delta and parts of the San Francisco Estuary. Since 1975, the environmental monitoring program has routinely collected discrete water quality, nutrients, phytoplankton, zooplankton, benthic samples, as well as continuous water quality data. The extensive monitoring has established a baseline ecological record of the Bay Delta ecosystem, which has been crucial for effective management of water resources. In addition, it's also part of IEP's monitoring work component. So the primary objective of the EMP is to provide data on the effects of river flow regulation and water diversions on water quality. This ensures the maintenance of water quality standards specified in California water quality control plans and water right decisions. Water right decision 1641 or D41 is the latest regulatory compliance document that establishes these water quality standards for the State Water Project and the Central Valley Project. And it mandates that the it mandates that DWR, USBR, and other state and federal agencies conduct comprehensive environmental monitoring programs to ensure compliance with these standards. I've listed the standards right up here, and these standards are required to be maintained at the various stations listed in Table 5 of D1641. So the environmental monitoring program consists of two monitoring groups. One is the discrete EMP and the other is the continuous EMP. Discrete EMP collects monthly water quality data and water samples for laboratory analytes, typically on board the Sentinel at fixed stations throughout the Delta. These data are made available through online platforms like the Water Data Library. And then the continuous EMP, on the other hand, provides quarter-hourly real-time data at fixed stations, and that's relayed to online databases such as CDEC. You can find the information there. So the discrete water quality monitoring has been a crucial component of the work conducted by DWR and USBR with assistance from the California Department of Fish and Wildlife. Their compliance monitoring includes 21 fixed stations throughout the Delta. Some most are accessed by vessel or the Sentinel, and a few of them are accessed by vehicle. So they're shore-based stations. At each station, a vertical YSI XO2 multi-parameter SON is deployed to collect various water quality parameters at both the surface and the bottom of the water column. And we also collect water samples and send them off to a laboratory for numerous analytes, which include dissolved nitrate, nitrite, total phosphorus, and total and dissolved organic carbon. Sampling consistently occurs at high slack tide every month or every day when we go out, and it's primarily conducted using the Sentinel. And these are the 18 vessels based stations, and 3 of them are shore based. And beyond water quality monitoring, the discrete EMP also performs monthly sampling of phytoplankton, zooplankton, and benthic invertebrates. And collectively, these data provide a monthly snapshot of the water quality conditions and biological communities, which are essential for documenting long-term ecological trends and informing management and operation of California's water delivery systems. So in addition to its mandated monitoring, the discrete also engages in scientific collaborations with other state and federal partners, as well as scientific or academic institutions. We've provided resources to collect data on harmful algal blooms, helped with developing taxonomic identification keys of zooplankton cultures, and some other important pivotal research efforts. In its pursuit of scientific excellence, the discrete EMP continuously seeks to improve sampling methodologies to ensure the data is collected accurately and meets the latest scientific collection standards. And one of its key advancements that we've implemented is the use of the Sentinel's onboard flow-through system. This requires an additional YSI XO2 multi-parameter SON and also a fluoroprobe. You can see kind of in the picture on the far right side, they're hooked up there on the boat. And this allows us to record water quality parameters and chlorophyll concentrations continuously wherever the Sentinel navigates throughout the Delta. And the initial or primary purpose of this additional SON, which we refer to as the horizontal SON, was really to just compare the data that we were collecting from the vertical SON and help validate and verify the data that's being collected. All right. So to capture this data, Discrete uses a software called Moped, which is a customized data logging software developed by DWR staff. It's integrated with the Sentinel's onboard flow-through system and collects water from 1 meter below the surface and delivers it to the horizontal SON. It can connect to both the vertical and the horizontal SON so that it simultaneously collects data while the Sentinel is navigating, and also when we stop at each station to collect our surface and bottom data. Moped is engineered to continuously record measurements at 5-second intervals, and it integrates with the onboard GPS system, tagging each water quality record with precise latitude and longitude coordinates. This results in a substantial data set of spatially explicit water quality data that can be used to generate detailed spatial maps of the surface parameters in all the areas covered by the Sentinel. You can see in the blue line there. So this project really, the inspiration for this project stemmed from recognizing that this data, this comprehensive data set that is collected every time we go out in the boat has a lot of value, potential value, depending on how you want to use it. The data set comprises over 100,000 spatially explicit data points and covers a wide geographic range of the Delta. And this extensive coverage from this high resolution data provide a robust foundation for various scientific analysis purposes and management considerations in the region. However, before this project, this data was only available internally or upon a request. The objectives of this project were to adhere to the fair data principles so that the data can be findable, accessible, interpretable, and reusable. So that allows us to confidently publish this data. In the data set right now, we have data from 2017, annual records from 2017 to 2024, but it's intended to update the data set every year with the latest horizontal moped data. It includes 7 standard water quality parameters, which include water temperature, salinity, specific conductance, pH, turbidity, chlorophyll fluorescence, and dissolved oxygen in both milliliters, milligrams per liter and percent saturation. The data set has been limited to 1-minute intervals to reduce noise and improve any analyses, but more granular data could be available upon a request. And then each record, again, is tagged with precise latitude, longitude coordinates. So I just want to take some time to explore the data set and illustrate some of its potential use cases. I've selected a subset of the data from our January 2024 run and plotted 6 of the parameters in ArcGIS. Each parameter is represented with a color gradient where higher measurements are in darker shades and lower measurements are in lighter shades. And you can observe some distinct but potentially expected trends in some of the parameters like water temperature or specific conductance or chlorophyll fluorescence. And although these visualizations are just static snapshots of the region, of the single monthly run, they do provide valuable insight when you compare a month to month or over several years. And that really just depends on your type of analysis. Our goal in providing this data set is to allow users to filter and analyze the data according to their needs, and to really not have any inherent bias in the data set that is provided. It's also important to note that this data set is not designed for optimized coverage of the Delta. This information is simply just complementary information that we get from the Sentinel as we go out and drive around on our monthly cruises. So with that same data displayed in the previous slide, I've zoomed in on an area here in the Central Delta and used some geoprocessing tools as examples to further analyze the chlorophyll fluorescence data. On the left-hand side, I've used the generate tessellations tool to average the data points within each cell. This method involves dividing the geographic area into smaller, non-overlapping shapes like hexagons or squares, and creating a grid on the map. It aggregates the data within each cell either by averaging or taking the sum, max, min, standard deviation. There's plenty of methods. And it provides a discrete representation of the spatial data, which can make it easier to analyze relationships. On the other hand, I've used the same data to interpolate the results, which involves estimating the values at unsampled locations based on the known data points. So this method creates a continuous surface and it predicts the values in the areas where the direct measurements are not available. So just want to touch on a couple examples of how this data has been used in the past. And so this is more specific to the Delta. This study was conducted under the auspices of the IEP and a special issue on the effects of drought on the ecology of the upper San Francisco estuary. The study investigated cyanobacterial phytoplankton blooms using data from 2014 to 2021 to illustrate how flow dynamics, water temperature, and water clarity can influence the occurrence of microcystis. This analysis incorporated horizontal moped water quality data and our fluoroprobe data that was on board the Sentinel. And to really make this all come together, the fluoroprobe data was spatially aligned with the GPS tagged horizontal data. And then the results here were interpolated using ArcGIS, the spline with barriers tool, to create a continuous map of the values across the map domain. And this result demonstrated how using modern in situ monitoring data improves spatial resolution, allowing for better exploration of the relationships between environmental conditions and the formation of harmful algal blooms. Another study, or another way to use this data is potentially for habitat suitability maps or habitat suitability analyses. This study looked at habitat suitability analyses for spawning adult Delta smelt. It compared habitat suitability during a dry year in 2008, I think, and a wet year in 2006. And to really understand how maybe the variations of presence or probability of presence in Delta smelt occurrence might change. This project developed a habitat suitability model using some of the fish survey data and integrated it with the water quality parameters such as secchi depth, turbidity, and water temperature. Although this study did not conclusively explain the reasons behind the observed changes, habitat suitability analyses nonetheless provide a comprehensive understanding of aquatic ecosystems interactions between physical, chemical, and biological factors. So those are just a couple examples of how you can use the data. I really wanted to have this data published on EDI before this presentation, so you're distracted now with the cat meme. I am in the process of just wrapping up a couple QA/QC tasks, and I do anticipate this being published on EDI within the next couple of weeks. So if you are interested in this data and you want it sooner than later, please feel free to reach out. This is my contact information. And then, yeah, in a couple of weeks, it should be available on EDI. So that's it.
Have 5 minutes for questions. So we'll start with anybody in the audience have a question? Okay, anybody online? Oh, there's one over there.
Hi, great job. Emily Richardson, USGS. We collect similar data and it's usable for roughly 1 million different types of studies. So really great job publishing this. I was wondering if you were also going to be publishing your FluoroProbe data.
The FluoroProbe data is already published on EDI.
Oh, great.
Yeah. So you can find it there.
Technical question for those maps, do you go out during a 1 week period? And so you're integrating data from like a 1 week snapshot? And how are you handling the overlap in the data set if you're going out to some areas of the Delta 2 consecutive days or something like that?
Yeah, that's a great question. So it does have a few caveats. So over a 1 month, what I showed in the GIS or in the example, we go out over 7 days and to different parts of the region. So for the most part, the overlap is in that data set. And it's one of those things that whoever wants to use it or how you intend to use it, you will notice that. But I'm not doing any specific analysis purposes, so there is quite a bit of overlap, especially around the area where the Sentinel is in the marina, so where we have to come back every day. But that is definitely a caveat to the data.
Other questions? Okay, we'll move right along. Thank you, Greg. Our next speaker is Morgan Gilbert with CDFW.
Thanks, Adam. I was actually having to do a little bit of mental math. Fun fact, I started at the Lodi Fish and Wildlife Office the same day Adam did. And so when he was talking about being there for 8 years, it was like, wait a second, what's going on? But good morning or good afternoon. I'm here today to talk to you about the larval entrainment study, which has been running since 2022 alongside the smelt larval and 20 millimeter surveys. Many of you are probably already familiar with the logic behind our study. So today I'm going to take you on a tour of our 2024 season, including some snapshots of the analyses that we're working on, investigating the effect of catch depletion, special study on diel variation, and what our seasonal results are telling us so far. If you're interested in what I've been up to in our previous years of sampling, I gave a presentation last year that kind of runs through a lot of that and dives into the literature a little bit. Before I get to the sciencey stuff, I'm going to take a moment to call out the people that have helped make this study happen. I want to take a moment to thank our funders at the Department of Water Resources. And I'd also like to acknowledge the many collaborators that have contributed to this developing project, including the staff of the CDFW Native Fish Unit and Fish Facilities Unit, Water Branch, the Department of Water Resources, and the US Fish and Wildlife Service. Alongside them are the many people that have contributed to the design of the study or to our field work. We could not have done all this without you. So now I'm going to back up even further and introduce myself. My name is Morgan Gilbert. I'm an environmental scientist working for the California Department of Fish and Wildlife, and I work for the Fish Facilities and Entrainment Unit. And in fact, you could call me the E in FFE. And that's because I lead the larval entrainment study. That started out as LEPS, or the Larval Entrainment Pilot Study, back in 22. If it seems like I've got a little bit of a chip on my shoulder, it's because I now call myself a lesser instead of a leper. At least now the doctors tell me I'm no longer contagious. But we're focused on the problem of entrainment. And so just to make sure everybody's up on the vocabulary, entrainment occurs when fishes are diverted by man-made flows. Generally those originating from water extraction operations out of their preferred habitats into neighboring regions. There's some nuance to the kinds of entrainment that fishes might encounter. Those within that red circle there, down by the State Water Project, might experience direct entrainment, while those further out down in the Old and Middle River Corridor, or OMR, might experience more indirect entrainment. The question of entrainment and its effect on the fishes of the Delta is a problem that goes back a long time, all the way back to the 1960s when the facilities were first being constructed. I have here just a tiny vertical slice of the literature that's come out regarding the effects of entrainment. I'm just going to touch on two major points here. The first is that entrainment has been suspected as a contributing factor to the decline of several species, including Delta and longfin smelt. And while there has been a considerable modeling effort to understand what the impact of entrainment is on the population dynamics of those species, there does not yet seem to be a single consensus. And so that's what LESS is here for. We exist to kind of address that gap, give managers a better picture of the impacts of entrainment, and provide modelers with ground truthing and more data for future analyses. So with all that introductory stuff done, I can dive into our 2024 season. We made some big changes last year. And since then, I've had a little bit of a chance to dig into our data. This is going to be a high-level overview of our season, and then I'll dive into some of these special studies and kind of more in-depth analyses. The biggest change we made in 2024 was a geographic extension. We started out in previous years working directly adjacent to the State Water Project in West Canal. In 2024, we expanded to a series of SLS stations along the San Joaquin and Old and Middle River corridor that we believe would give us a good spectrum of indirect and direct entrainment risk. Two of those stations that I've highlighted here in blue, we designated as being our priority stations. These are sort of the core of our study. These were sampled 3 days a week. The first day of the week was an intensive round of sampling that we call the priority day, where we just sampled those sites with multiple replicate tows. And then we also ran what we called a transect run. The other 2 days of the week that we sampled, we would go out and run tows at our priority stations and a set of transect stations running all the way down to station 918 down by West Canal. Our aim with this expanded area sampling again was to provide a broader and more consistent picture of longfin distribution within the zone of entrainment. The other big change that we made in 2024 was the bongo sled. This gear is a dual net design, similar to an SLS sled, just sort of mounted together. And our initial testing with this gear was so successful, in fact, that we ended up using it exclusively after the first few weeks of sampling. Now I'm going to turn to talking about some of the analysis that I've been doing. But before I dive into our seasonal results, I wanted to throw up a couple of quick warning signs. I am still in the process of reviewing this data for quality control purposes, so some of these results are preliminary and provisional. Still, I think we're moving in an interesting direction. I hope you might agree. This is our 2024 season at sort of its broadest resolution. On the left here, I've got our mean catch per unit effort in fish per 1,000 cubic meters sampled. Our priority sampling is coded in red, while our transect sampling is in blue. And you can see that our catch was pretty consistent throughout the season, which started in January and ended in March. Catch peaked for us in early February. Both of these protocols collected roughly similar numbers of fish, but the vast majority of those were prickly sculpin, which is not unexpected for our system. It's not necessarily a bad thing. They are a native species. And while they did dominate our catch, we did collect 211 longfin smelt, so an order of magnitude smaller. Again, I've laid this out in fish per 1,000 cubic meters. So as you can see, we caught longfin throughout our 2024 season from some of our earliest sampling in January all the way through when we completed our sampling in March. Both our priority and transect sampling detected longfin. Our highest detected densities came in early February, which makes sense. It's when longfin hatch. We're planning on further investigating our 2024 season by looking at things like length distributions of the species that we collected, perhaps engaging in some community analysis and looking at various environmental factors. but I'm going to zoom in on a few of the analyses that I've completed so far. And the first one that I was interested in investigating is catch depletion. And that is because one of the things that sets LES apart from our sister studies is that we conduct replicate tows at a smaller subset of sites. So the concern we had was that these replicates might be depleting the number of fish that are available to be sampled, potentially skewing our overall CPUE downwards. And so to investigate that, I ran a regression between our catch per unit effort and the sequential tow number. We would expect to see if depletion was occurring, a relationship between those two. Nothing emerged out of that linear regression. This was using all of the fish that we collected. Even when we drilled down to looking at just longfin or just prickly sculpin, this relationship held up, or lack of relationship held up. This seems like a pretty reasonable result. We are carrying out replicate tows at these same sites in sequence, but we're not sampling some shallow little stream. This is a big, broad, deep river. And so fish seem to be present in sufficient numbers that depletion is not a major factor. Another thing that I'd like to highlight is that we completed a series of night-day comparisons as a special study this year. It's commonly understood that larval fish tend to be more active at night. And other studies have indicated that longfin follow that pattern. In fact, I'd like to plug, we have an article coming out in the IEP newsletter talking about our 2022 day-night comparison. Take a look at that. But in 2024, we decided to take another look at that and investigate what effect that sampling would have. So we completed 3 overnight sampling trips at our priority stations during 2 different weeks in February. You'll hear me talk about mid-February and late February here in a little bit. These sampling trips ran from about 8 p.m. to around 3 a.m. To ensure that we were sampling in total darkness. And I gotta say, this sampling took a lot of effort on the part of our crews. Working at night makes everything about being out on the Delta much more difficult. I was very fortunate to have some very experienced and extremely caffeinated boat operators. So this is the night-day study kind of at its broad resolution across all taxa. We found that we did indeed have our highest overall catch at night, which I've got highlighted here in blue. Prickly sculpin were again the dominant taxa that we collected. And our catch was highest during our mid-February sampling and somewhat lower during our sampling later in the month. We did not actually collect any longfin during our first week of overnight sampling at night. We did during the day, but we did catch them in some numbers during our second week of sampling. We consistently collected longfin during our daytime sampling, with the highest overall catch coming during our daytime sampling in the mid-February trips. We had planned on an additional sampling day in the early part of the month, but bad weather resulted in us having to cancel that. So with that in mind, I set out to investigate what differences existed within this data set. To answer that question, I made use of a Permanova to compare our day and night samples alongside other factors that might be influencing our results, specifically the station that we sampled. Remember, I was doing 809 and 812, and the week that we sampled. So perhaps a little bit surprisingly, the diel period that we sampled did not seem to have a significant effect on our catch, and neither did the geographical region that we were sampling. What did produce a significant effect with this data was the time period that we were sampling in. That is, whether it was collected during mid-February or late February, which explained about 23% of the observed variation between our samples. Here I've got ordination. This is a non-metric multidimensional scaling. And as you might be able to make out, our mid-February samples, the little triangles here, have a pretty distinct clumping pattern on the first axis, separating them from the more sort of widely distributed later February samples. There otherwise does not appear to be any other strong associations within this data set. So with these analyses in mind, the outcome of the special study seems to indicate that when you sample, i.e. seasonally, that has a greater effect than the time, i.e. the time of day that you're sampling. Turning back to our 2024 season results, if seasonal timing was the primary thing shaping our day-night sampling results, what about our sampling season as a whole? Once again, I made use of a Permanova to investigate this. In this case, I made use of all of our 2024 season data, excluding our special studies. And in this case, I used the matrix opposite of 4 factors: the month that we sampled, the Julian week that we sampled, the station that we sampled at, and the protocol that we operated with. That is, whether it was a priority day or a transect day. While testing produced significant p-values for the month, week, and station sampled, if you peek a little closer, you can see that the R² for all of those is very low. So basically, this is the Permanova telling us that our results are significant but insignificant in a way, and that they're not really covering much of the variation. And this very high residual R², about 0.89, indicates that about 90% of the variation within this data set is not explained by any of these factors. So while it seems reasonable offhand that our seasonal timing and geography might be significant to our results, they don't seem to be the primary factors shaping this data set. So we're going to need some further analysis to understand what exactly shaped our 2024 season. And so where does that leave me? Well, we're a work in progress. The best is yet to come. I'm excited because we're just sort of barely scratching the surface with these analyses. Wrapping up this early exploration of our data set, I should note that we have a lot more coming than I could hope to do justice in the short time that I've got with you today. I haven't talked about our 2025 season at all, and that is going to include more changes and improvements to our sampling design, including the addition of a new site that will provide us with a downstream control station and let us do some occupancy analysis. Along with that, we're planning on not just completing, we're working on a special study to assess the vertical distribution of longfin smelt within our sites. And we're also collaborating with the DWR Genetics Monitoring Lab on their eDNA study and have been sampling alongside them. So we're excited to keep growing alongside all of you. This is sort of a snapshot of us in progress. And that's the end of my talk, more or less. If I've got time, I'm happy to take any questions. Right here in front. You can just yell too. So we conduct oblique tows. Sorry if I didn't make that super clear. We sample using a similar set of methods to the smelt larval survey. So it's based on the depth and then the amount of line that we let out. We sample anywhere between 60 feet of water at the maximum to maybe 15 feet in our really shallow sites.
So how would you expect to see a difference, a DL difference if for instance, they're migrating within that depth range?
So the tow we make use of is an oblique tow, right? So at the start of the tow, we're dragging the net at its maximum length and it's close to the bottom. And then we draw that net upwards at basically a set of time increments to sample the whole water column. So one of the things that we're looking at with this comparison study that I was talking about, the special study that we're looking at this year, is just looking at what if we just tow at the bottom? What if we just tow at the surface? How does that affect our catch? What are we seeing when we switch that up? And so that's one of the investigations that we're setting out to look at.
Anybody else have questions?
Must have been a good talk. Thanks, everybody.
Thank you, Morgan.
Awesome.
All right, our next speaker is Jennifer Witt. She's a supervisory fish biologist with the U.S. Fish and Wildlife Service.
Hi, everyone. Yeah, as Adam said, my name is Jennifer Witt. I'm with the LODA U.S. Fish and Wildlife Service here in California. And today I'm going to kind of build on a little bit of what Adam was talking about earlier with our Delta Juvenile Fish Monitoring Program, also known as DJFMP, and our beach scene expansion project. to quantify the relative abundance and distribution of juvenile Chinook salmon and other co-occurring fish species in our nearshore habitats, the DJFMP has conducted beach seining in the Lower Sacramento River, the San Joaquin River, the Delta, and the San Francisco Bay since 1976. In 2013, the DJFMP underwent a programmatic review led by the IEP Science Advisory Group. The review resulted in recommendations to measure the efficiency of capture using beach seines and random site selection to enable calculations of absolute abundance. In 2022, the DJFMP initiated a beach seine site expansion project aimed at strengthening standardization sampling procedures, eliminating selection bias. Today, I will provide a brief history of the DJFMP seine program, discuss the approach taken for the site expansion and selection, and report some preliminary successes of the project. Throughout the years, the DJFMP has seined 73 different fixed beach seine sites, which gradually changed over time due to changes in the fluvial systems. The beach seine sites are stratified into 7 geographic regions and seined once weekly. These are the Lower Sacramento River, North Delta, Central Delta, South Delta, San Joaquin River, and the San Francisco and San Pablo Bays, also known as our East Bay and West Bay seines. So that our bay seines are sampled once per week, or alternatively. Sacramento seine sites are sampled once per day from October to January to target our winter and spring runs Chinook salmon. So why expand the beach seine sites? Well, it's because fluvial systems change, right? Even in the calmest of years. Rivers meander as snow melts, spring rains fall, and soil deposition occurs. The majority of the delta has also been altered via anthropogenic changes such as channelization. These changes have opened the system up to introduce species on a large scale. As of 2023, non-native species comprise nearly 75% of the total submerged aquatic vegetation cover in the Delta, Chrisman et al. 2023. This equates to some loss of our sampling sites due to this SAV encroachment. The beach seine site expansion project allows DJFMP to complete more seine site sampling in each region by incorporating more new sites into that same list. This expansion, along with DJFMP's beach seine efficiency project, will also help the DJFMP Special Studies Program, led by Brian Mathias, calculate the abundance of species at each seine site at different times of the year, rather just our CPU trends over time. So how do we kind of work this out? In 2022, the LFWO, spearheaded by Adam Nanninga and Mike Marshall, began transversing the Lower Sacramento River by boat to look for possible new seine sites. During these scouting events, the team located 24 potential new seine sites in this LS region. It's important to note here that we actually it's not feasible for us to actually seine those sites by boat, as this is a 2-hour drive each way. And there are parts of our river, of course, in Sacramento that are just impossible to pass. Then what happened in the winter of 2022-23, a series of storms hit, as we all know, and it dumped record-breaking amounts of rain and snow across California. This put our beach seine expansion project on hold as other pressing issues and safety concerns for the team surfaced. Even as the waters receded in the Delta, the amount of sedimentation and debris left behind had altered many of the fixed sites in our regional runs. In the fall, however, of 2023, we were actually able to revitalize the project, and we were able to mail out 22 letters to different landowners who had properties that were adjacent to those sites that we looked at. So of these 22 letters, 11 of them were actually returned. Of those, 9 were marked yes, and 2 were marked no. This allowed us to actually contact these landowners. Some landowners met with us on site for further discussion and to help us evaluate the locations. Others granted us access over the phone. During our site visits, we assessed easy accessibility and whether the sampling sites would meet our needs of the DJFMP program, noting that the previous year's flooding had altered much of our landscape. Despite these changes, we successfully added 3 new SANE sites to this run. Our next step involved reestablishment of our scouting teams, and within 3 weeks, we located and contacted landowners in all 5 other river and brackish water regions. We also reviewed our current standardized operating procedures, our SOP, and our best practices. This enabled our sampling teams to expand their search for alternative sites when the fixed site was not viable for seining. In 2024, after extensive program evaluations, we revised the selection criteria to include factors such as distance from the fixed site, and along with compatibility. This update to our SOP also facilitated the identification and establishment of our new sites in each region. So some of our preliminary data shows us that you can see here in our gray line, that's actually our current, the 2024 calendar year in this slide. And so if we look at the difference between those, the orange is actually 2023 and the blue is 2024. With our gray line, it is important to kind of note that you can see our first major kind of incline in our sampling ability happened right around where you would see that first quarter, that February, when we allowed our teams to go out there and sample a little bit more with that fixed site expansion part of that. Then, of course, in that fourth quarter, you can see that growth that we had again in our sampling near the end. With this one, we have our little pie charts. Our first number is the amount of completed samples that we did in each region, along with the percentage of the sampling that we did. I think that with our preliminary views, it's pretty impressive that with most of all of our regions, in fact, we were able to complete more of that sampling for each one of those. The only one that is a little bit down would actually be our SAC-SANE, which is in that October to November time period. And that one doesn't have any extra SANE sites. So we had no ability to do a randomized sampling through those. Those are fixed and those are set for those. So my conclusion is the associated team continues investigating alternative sample sites to maximize data collection opportunities. Since the start of 2025 water year, the DJFMP seining program has added 46 new SANE sites to the existing 58 historical sites we've been sampling. This expansion enables the program to select 8 sites for each SANE sampling run randomly. The Beach SANE site expansion project demonstrates that even minor modifications in monitoring can significantly enhance the utility of the collection data. And of course, my big takeaway is partnering with stakeholders is worth the effort.
I forgot to mention at the beginning, this is a lightning talk. So
I can talk as fast as I could. You guys are let me know. I can talk.
the 8-minute talk. We do have time for questions. Don't go too far.
Okay.
Questions about this presentation? Anything online? No? Okay, thank you, Jennifer. We're a little ahead of schedule, but I don't think anybody will complain if we just keep rolling. Our next presenter is Margot Matson. She's a California Sea Grant Fellow working with the Delta Stewardship Council in the Adaptive Management Unit as Delta Independent Science Board staff support.
Thank you. Hi, everyone. I know this is the last presentation before lunch, so thanks for hanging in there. My name is Margot. Today, I'm going to talk about a new proposed review by the Delta Independent Science Board that will focus on contaminant monitoring. And our current working title is Contaminant Monitoring in the Delta to Inform Ecosystem Management. So as many of you know, the Delta ISB, or Independent Science Board, was formed by the Delta Reform Act in 2009 to provide oversight of the different activities in the Delta and monitoring and programs to support adaptive management and provide independent advice for implementation of the Delta plan. And one of the ways they do this is by conducting periodic reviews on programs and major challenges that are facing the Delta. So one of these major challenges that this particular proposed review is going to focus on is this continuous inflow of contaminants into the Delta. So under the Clean Water Act, the Delta is considered an impaired waterway, meaning that it's exceeding these allowable total maxima of daily loads of contaminants, urban and industrial effluents, or chemical contaminants, metals, insecticides, and toxicity. And so a lot of these contaminants are entering the waterway through agricultural runoff, urban runoff, wastewater effluents, and industrial effluents. It can be really difficult to assess the toxicity and risk of these contaminants. Some are known to pose ecological threats. Some that are more of these new emerging constituents of concern have unknown threats to the ecosystem. And it can also be difficult in the environment because there's interactions between chemicals and contaminants, but also interactions between these contaminants and different environmental stressors like drought and temperature. So current approaches might be overlooking some of these significant sublethal effects that contaminants could have and potential food web level consequences of contaminant exposure. So to address this challenge, the Delta ISB is proposing this new review that will look specifically at contaminant monitoring in the Delta. And it will be building upon previous work that the Delta ISB has conducted, namely the 2018 Water Quality Review. And that review focused namely on contaminants, nutrients, algal toxins in the Delta, and how this data and research can either be used or might be ignored in some decision-making that was happening. In the 2018 review, monitoring wasn't thoroughly covered, as it was also addressed in the 2022 Monitoring Enterprise Review that gave a pretty big overview of different monitoring efforts in the Delta and assessed the ability to support adaptive management. And some major conclusions were that it's unclear if and how this data is entering into decision-making, specifically around flows and water storage and how that can influence water quality. So there's a need for more holistic studies that can assess contaminants, and maybe this chemical by chemical approach might be insufficient considering the effects of mixtures and multiple stressors in the environment. And as noted by the 2024 Delta ISB Food Web Review, there's also this need to understand the flow and the consequences of contaminants within the food web. So the proposed review has three main goals. The first is to review and assess current monitoring programs in the Delta. And so this is asking questions like, are we monitoring for the right things? Is the list of analytes that is currently being used up to date based on the different identified sources of contaminants that are suspected to be entering the system? Are we monitoring for any outdated compounds? Are we missing any key compounds that might be monitored for in other systems that are known to pose ecological threats? Also, can these programs adequately assess ecological risk? Are the current methods and approaches sufficient? Is the spatial and temporal scale of the monitoring sufficient? And have recommendations from previous efforts that have looked at monitoring programs, constituents of emerging concern, and risk assessment been incorporated into current monitoring programs? Also, if programs are ecologically relevant for the system and these different stressors that we have at hand. The second goal of the review is to understand how monitoring can better inform decision-making. So we hope to look at what regulation is driving this monitoring, where is the funding coming from, how flexible might these programs be to change if new constituents are identified or there's new sources of contamination. What are the needs of managers, and what are any triggers for management actions or for changes to monitoring programs? And how can this data be better integrated, incorporated into ecological models? The third goal is to identify any shortcomings of programs and propose solutions. So this would be to identify gaps in current monitoring programs, understand if there's any barriers or obstacles that exist to creating a more ecologically significant program in the Delta, and learn from other systems and countries to see what approaches are being taken that could possibly be implemented in the Delta. So thinking if we can move away from more of a chemical by chemical based approach that might fail to represent some of these larger environmental conditions to something more holistic. And so the review approach will include 4 key phases. So the first will be interviews or scoping discussions with key experts, and this will help understand any gaps or barriers or obstacles. We also hope to gain insight into more of the regulatory landscape that's driving monitoring and explore how these management decisions are being linked to monitoring programs. The second phase will be a review of reports and literature. So reviewing what chemicals are being monitored, what are the different methods being used to assess risk, understanding any past reviews and recommendations to see if they've been implemented. The third phase will be to explore new tools and approaches and to see which ones could be implemented into the Delta. And so this will be exploring what else is out there, what's being used in different systems with success, what other types of methods that we could incorporate. And phase 4 is sort of an educational sharing back or learning opportunity. So it's our seminar series. Hopefully we can talk about some of these tools that we might have identified. It could include topics like ecotoxicological tools for risk assessment, effect-based methods like bioassays, predictive modeling, and non-target chemical analysis. So the point of this presentation is really to just let you all know about this proposed review. And we're soliciting feedback if people have ideas or comments that they would like to provide. We are working on getting out a draft of the prospectus for public comment. And in the next couple of weeks, you should expect to see it circulating on the IEP mailing listserv or also available on the website. So you're welcome to write us at the or the email address that's listed here, or also Dr. Inga Werner, who's sitting right here in the front row. She's going to be, well, she's the Delta ISB chair, but also the lead of this contaminants review. So you can also talk to her about any questions or comments you have on the proposed review. And with that, I will finish them. Thank you.
Yes, we have about 3 minutes for questions.
Hi, so how does this interact with the Delta Regional Monitoring Program and the plans that they're already making in their work plans? I assume they would at least be some of the key experts, but just curious about any prior coordination.
Yeah, I don't know, Inga, you want to answer?
Hi, and thank you, Margot, for a great talk. Of course, we will be talking and learning from those people. And this is obviously the major contaminant monitoring program in the Delta right now. So yes, this will be a major focus. And I'm looking forward to learning from all the experts who have put that together, of course. This is not, I just want to say this is not, we're not approaching this with the intent of make it better. We just want to review and see all the information and the thinking that went into the current program, understand the structures, understand what the managers need to actually use that information, and bring in maybe a few new tools or knowledge about new tools that could be used to get closer to the ecosystem effects that we all want to know about. So that's the brief one.
Probably one more question.
Okay, thanks, Miguel.
All right, it is now time for lunch. Thank you all very much all the speakers this morning for an excellent morning. If you are participating in the mentor lunch, that will be taking place on the 4th floor. And you need to be escorted up by someone with a badge. So please meet over on the other side of the lobby by the security desk in about 5 minutes, and we'll walk you up, or find me if you have issues. Other folks, feel free to find some place for lunch nearby and be back here at 1:50.
Go ahead and get started. Our first session after lunch is Muddy Waters, Clear Goals, Restoring Wetland Ecosystems. Our first speaker is Dan Ellis from the Interagency Ecological Program.
Everybody can please take a seat. We're going to start the session. Okay, we're going to start the session. So thank you, everybody. As Jenny said, my name is Dan Ellis. I'm with the IEP synthesis team. And today I'm going to be presenting some preliminary findings from a pilot study on primary productivity that was carried out at some of the fish restoration program wetlands. I first want to start by thanking the multitude of people who took part on this project. This is a true IEP success story because many IEP stakeholding groups were involved. We ended up pulling this study off for less than 10 grand in cash and 9 field days. So, but specifically, I want to call it the Fish Restoration Program at DWR. They provided boating support, logistics. They set up sondes at some of the sites we needed. They analyzed our water samples. The Fish Restoration Program at CDFW also provided boat time. They helped design the study and provided vegetation maps. USGS showed up with a new tool that might have adaptive management potential so that we could collaborate and compare our results at the same time. We had colleagues over at UC Davis running the incubations, providing access to their facilities. We got funding through a NASA grant with UC Merced, and I also have to thank my colleagues in my group. So the purpose of this study was to fill foundational ecology questions we have about wetlands. Something as simple as, how do we support, or how do these wetlands support the base of the food web, and abiotically and biotically? And then can we design a wetland to optimize for those benefits? But that brings me to the first key question. How does one correlate an ecological outcome with a specific wetland? And so here's how we do that. We looked at the water over a flood ebb cycle. So the water flowed onto the site and the water flowed off the site. At each site, we had at least one set of continuous sondes deployed at the entrance to the wetland. And those were measuring for depth, temperature, conductivity. Some of them had FDOM, pH, DO, and chlorophyll. At a wetland like this, it's fairly simple. The majority of the water that flowed in through that entrance flowed out through that entrance. But not every site is the same. So this is Wings Landing. This is a restoration site very close in proximity to Rush Ranch, which I just showed. And you can see there's multiple breaches. Water that enters is not discretely going to enter and exit via the same box. So the black box flood minus ebb kind of premise isn't as strong at a site like this. These sites are really complicated, and this is expensive work to do. So obviously you have to test out these methods. For each of the sites I'm going to be showing you, we went out during daylight hours and collected 3 times during the flood and 3 times during the ebb. And we collected a number of different discrete samples, but remember again, all of these sites were equipped with continuous setups as well. The sites we looked at were broken up by region. There's multiple regions of the Delta that we find important for our listed fish, so we wanted to make sure that we were understanding the system in different locations. In Sassoon Marsh, we looked at Wings Landing, which is a restoration site. Rush Ranch is a reference. We also looked at Lower Yolo Ranch and Yolo Flyway Farms. The general premise here is that more or less the same water is going in as what came out. Or sorry, rather the same water is going into both sites. So whatever differences we do observe have to be due to something about the wetland. And then ideally, can we manage the system based on what we found? We focused in on the flood ebb cycle as well as some seasonality. We went out to each site every 2 months, and our last sampling event is actually coming up in April. To a lesser extent, we did touch upon the spring neap cycle, and this is really important. But this was more haphazard for us. We didn't touch upon the interannual cycle. Sampling looked like this. We would go out to a location and we would bop back and forth all day, nonstop, between the entrances to the 2 abutting wetlands. And so on a given field day, you would do 2 sites in that same region and all day, just back and forth collecting water samples. Water is getting collected in the same format as what USGS does with their flow through system. We had a Y valve juncture, so raw water was getting split off. Filtered water was going directly into sample containers for those analyses. And we collected a lot of data. I've grayed out phytoplankton because while we have a sample for every measurement, for every event, we don't yet have the funds to analyze those samples. So we're just storing those for now. The Bright Labs deserves a lot of thanks. They have an enormous amount of work to do, and we had them analyzing our dissolved and total nutrients. We also did total suspended solids, dissolved and total carbon, chlorophyll. We really made use of what the Fish Restoration Program and DWR made available to us. This is a single day's sampling with 2 people. Those of you who collect this kind of water know that's a lot. So thank you to the Bright Lab people if they're listening. There was a second data stream as well. The water grabs that didn't get used for water quality analysis were sent off for incubations at the Cabo facility. Now, those incubations were about a 12-hour period, and a single sample would be split into 3 replicates. Each replicate was then split into a light and dark tank. So from the sample water I collected, that was 1 grab, it would be split into 6 containers. And that was because you need to calculate to get to the gross primary productivity, which is what I was looking at mostly. That's the amount of carbon that was acquired in that time span. You first have to understand what the net primary production looked like and the respiration. To acclimate you to the sites we looked at, I'm going to show you a conceptual diagram here. So in these bounding boxes, this is more or less what the sites represented in terms of site design. So there were dendritic sites. There are incised sites. Think of these as those were on a tidal cycle, the floodplain isn't inundated, but the channels or maybe some ponds are. And then there are sites that are subtidal, but they do have underlying geomorphology. For all the data that I'm about to show you, it's going to use this set of habitat categories. So this was aerial imagery provided by DWR, really high resolution drone photography. We split up into riparian or roads or levees. We called something the high floodplain. So what that means is that this was seasonally inundated, but typically dry. Then there was the mid floodplain. I like to think about this as it's not tidally active, but the vegetation have wet roots. So water's still permeating. Many of the sites, because of their old use, because they had been restorations, have what I've come to call capillary channels. Often they were irrigation ditches. These are shallow ways for water to move around the site that weren't necessarily designed or constructed that way, but they perform a lot of functionality. And then we obviously have the low floodplain or swales. Sometimes these were intentionally constructed, sometimes these were naturally formed. And then we have interior and exterior channels. So jumping right in, our first site is Yolo Flyway Farms. This is in the Cache Slough complex. And for all of the figures to come, this is going to be the colors assigned to those different groups, but it's also written beneath. Now, earlier I showed you how water flowed at Rush Ranch and at Wings Landing. Right now, I'm going to demonstrate what water flowed looked like at Yolo Flyway Farm. So while the site as a whole has its scale, the actual portion of the site that's represented by the data I'm going to show you looks more like this. So water flows into the site. It has the potential to expand out into a couple swales, into some channels, and at some point then the water is going to flow off. So at the site level, Yolo Flyway Farms has a good amount of high floodplain with roughly equal parts mid and tidal swell, as well as some channels. At Lower Yolo Ranch, which you can see there is actually connected to Yolo Flyway Farms, so really similar water source. I drew this circle here, or ellipse, as the bounding box. And this is what the delineations looked like there. And again, just to give you a sense for, on the left, I'm showing you the breakdown of habitat categories site-wide. And I even included portions of Lower Yolo Ranch that weren't within the restoration boundaries. But you can see that the portion represented by the samples I'll show you is quite a lot more of the tidal swale and channel habitat. And I should say, during the study period, we did not have floodplain inundation. So all of these, the general rule of what goes in comes minus what comes out, it was relatively true. So Rush Ranch, very different. Now we're in Sassoon Marsh. The majority of this site is tidal swale. And here is what that site looks like delineated. And I want to just draw attention to this, and I'm curious if somebody has an answer. We always talk about Rush Ranch as a reference wetland, one of our few remnant ones. But when I was delineating, I was able to delineate down to literally less than a foot wide channels. There were many linear channels and channels meeting at right angles in multiple places. So that to me looked like maybe some old use that maybe we aren't familiar with. And then here's Wings Landing. I bounded here the area that is probably best represented by the water samples I'll be showing you. And this site has quite a lot of tidal pond. If you ever go out there, you can access it. There's a kayak trail. Tons of birds make use of this site. If nothing else, this is a poop hotspot. So the way that these wetlands perform is not simply just the wetland itself, it's the kinds of organisms they bring into the party. And this is just to show you a little bit better of the delineations. I want to pause and just show you how detailed we got. Like I said, some of the channels I was bounding were as little as half a foot wide. And that's because of the quality of data that the fish restoration program is collecting and many manpower hours. So just real quick, I'm just going to give this to you before I jump into the incubation data side by side with the habitat characteristics. So you can see that Yolo Flyway Farms and Lower Yolo Ranch are pretty similar on a proportional basis, but then when you look at the acreage, Lower Yolo Ranch is an enormous site. So even though proportionally you might think to yourself, oh, well, you know, there's less tidal pond represented here, the actual acreage of that habitat type is quite large. And then lower in the marsh, in Sassoon Marsh, those two sites didn't match quite as well, and they had different morphology. So there's multiple factors there that could control the results you'll see. I want to point out here that we didn't even know if we could differentiate on a tidal cycle. So obviously we anticipate that there's probably some difference between what flowed on and what flowed off, but can we actually quantify that? We were able to do so with 3 samples. I thought that was pretty impressive. Now, obviously not at all time points, but you'll see for the rest of the data that it was very promising. So here at Yolo Flyway Farms, there was one instance where there was pretty clear delineation between the ebb and the flood tide. And I want to pause on this too, because the measurements I'm showing you are representative of what the primary production was from a grab sample. This wasn't water that spent time on the wetland. We took it before it entered and then thereafter exited. So if the water is more productive, it's probably because it picked up nutrients while on site. And if it's less productive, it's probably because it maybe used up nutrients while it was on the site. So there's really no good or bad in terms of that. It's just learning how the wetlands work and then potentially leveraging that to our, management goals. The next site was Lower Yolo Ranch. Here we had wider spread in the data. I think that's not too surprising given the size of the site and the variability of habitats within. And I'm not going to stay too long on each one individually because I will show you them all at once. Rush Ranch here, we actually do have more data that didn't make it into this data set. We had less delineation between what flowed in and out, but there are some really interesting hydrological things going on at Rush Ranch that I'm excited to talk to some folks about. And here at Wings Landing, again, very close to Rush Ranch, we saw more separation, but this site, that black box of what goes in minus what comes out was not as clearly defined. Okay, so here is all the data all together. You've got the delineations. You also have the actual incubation data. So right here, I think it's pretty clear that, for example, if we were to get 4 samples, I think it's really evident we would be able to differentiate flood and ebb tide. We actually did pretty well with just 3. And then I want to point out Lower Yola Ranch, Yola Flyover Farms. The first sampling event there, you can see that the colors, the ebb and flood swapped, but their water sources were nearly identical. So something about the sites, something that we potentially can manage for, led to that difference. At Wings Landing and Rush Ranch, we actually had something similar, but Rush Ranch, there's much less difference between what flowed and what came out. Maybe we can manage for something like that. Maybe we can design the sites for that kind of goal. And I do, sorry, this was just a reminder for myself. I do want to draw attention that the y-axis here is not the same. Okay, so big takeaways. We can differentiate tidal wetlands over a single flood ebb cycle. So water that's flowing onto a site, I can tell you that the water that flowed on and the water that flowed off from incubations can be different. We did this with 10 grand of cash plus a ton of in-kind contributions from all the different collaborators I mentioned earlier. And so I think this is a great example of what IEP can do when we're leveraging resources. And this was only 9 field days. So we've got a couple more coming up. And I'm really excited to see where the study goes for that. Then we also have that the flood minus the ebb values were inconsistent. So when we look at that, there's a lot of data to analyze, and it's great to know that, but I'm excited to know why, and to know mechanistically, can we understand that? And so that's where the next steps come in. So first off, we have another sampling event in April. Really excited to get those ones finished up and get some data collected. Also, the USGS went out with what's called a lab staff. It's a real-time primary productivity measurement device. It's been used in other systems. It's been used in the ocean. Can we validate it here in our system? These are the kinds of tools that, if we validate, could potentially be used as a real adaptive management tool for wetlands or for other portions of our estuary. I want to continue delineating the different wetlands around the Delta, hopefully automating the process. These manually delineated ones will hopefully act as the control for that process. We also have incredible vegetation maps. All the fish restoration program sites are delineated for their vegetative cover. And Aisha over at CDFW does that. Really excited to see what the relationships are between the site design and what vegetation was where. And in cases where the two are the same and there's some nesting, can we uncover what's really causing these primary productivity rates to change? We obviously have the water grab analysis. For those of you who go to the Bright Labs, you know that there's like a data delay right now that they're working through. I'm very excited to get the data to look up why we saw what we saw. Obviously, I have my suspicions. And then long-term, it would also be really interesting to tie what we saw to the continuous on data sets. So is it possible that between so Claren et al., had an algorithm where you could sort of estimate from a remotely sensed image the primary productivity, but can we do that at the wetland scale? My thought process is that maybe future wetlands are designed so that you have these SONs that are taking the same kind of data you guys are all used to, but we have them calibrated and tuned so we know what that means for productivity rates as well. And maybe you could use that as an adaptive management tool to guide your decision-making or a change that you want to make at the wetland. Depending on what we find, we might have more questions than answers, and that's where those phytoplankton samples will become really useful. Like I said, we don't have funding right now to enumerate those, but we're keeping those for about a year and a half, 2 years, in the hopes that we can use those to explain what we found. And with that, I just want to thank you for your time and for all the people who contributed. There's more than I can list, but thank you. Questions? No? Okay, sounds like we have time for a question or two.
Hi, super interesting stuff. First, were all of your sampling efforts conducted around just before and after high tide?
So we went out in the morning during like the rising part, like the beginning of the rising flood. So we captured it either in the middle and then into the end of the flood, and then fully through the ebb. Daylight hours obviously were an issue, and also, you know, enormously long days. So we did constrain it as much as possible. We tried to capture as much of the flood and as much of the ebb. Sorry, I keep on doing that. As we could.
Gotcha. And what's your thought on the possible influence of bird activity in these sites?
Yeah, so the bird activity thing came to me. I was actually at Wings Landing on an off day, and I got stuck out there. And if you do go, it's very easy to get stuck. And there were at least 400 or 500 birds. And we're all out there doing work. A lot of us are fish focused in our work, and we kind of don't think about those birds, but that is a lot of feces, and that's a lot of nutrients. And so some of these sites, just by having features that attract birds, might become nutrient sources just for that. So, yeah. Probably, yeah. Okay. I think we ran out of time. Sorry.
Our next speaker is Dennis Finger with the California Department of Water Resources. And just a reminder for folks online, you can submit questions via the question and answer button on Zoom.
Hi, everyone. My name is Dennis Finger. I'm an environmental scientist with the California Department of Water Resources. And today I'm going to be talking with you all about the Big Notch project, as well as upcoming operations and adaptive management studies going on in the Yola Bypass. So I'll start off with a little bit of an overview of the Yola Bypass itself. And I'll talk about what the Big Notch project itself is and how it came about. I'll go into a little bit of the construction status of the Big Notch. After that, I'll get into a little bit of the planned monitoring studies that are taking place and will continue to take place with the Big Notch in place. Along with that, I'll go into a case study of 2024 adult Chinook salmon fall run tagging in the bypass and get into some future implications of the project and of these studies. So to begin with, I'll start off with the Yola Bypass. So the Yola Bypass is a flood conveyance basin that begins with Fremont Weir up in the north that was built to divert floodwaters away from the city of Sacramento. The weir itself is a 2-mile long concrete weir that for the most part looks something like this most of the time throughout the year, but during certain winters when the river stage rises, it will completely inundate once waters get to an elevation of 32 feet, and it'll inundate the bypass. And a few notes there, it doesn't happen every single year. It's more sporadic, so 2 out of every 3 years on average, and maybe for just a few days or a few weeks at a time. But when it does, it can inundate up to 59,000 acres, so pretty substantial area. And initially, this was really seen as purely providing those flood conveyance benefits, and really was viewed as a sink for juvenile fish, juvenile Chinook salmon. But that kind of started to change with the work of a number of folks in this room or agencies in this room, including Ted Summer and DWR, UC Davis, and CalTrout, a number of groups that studied fish that were in the Sacramento River compared with juvenile Chinook that were spending time within the Yola Bypass. And what they really saw is that fish that were going and spending time in the Yola Bypass were getting larger and were growing faster than those that were in the main stem Sacramento River because of larger food availability within the bypass. So that paired with really an understanding of the ability for adult Chinook salmon and sturgeon to migrate upstream through the bypass really led to a paradigm shift in viewing this as being productive habitat and an area to improve, and that in turn inspired a number of regulations, some of which were talked about earlier today, that have called for habitat restoration for floodplain rearing habitat and adult fish passage within the Yola Bypass. So that kind of led to the project idea for the Big Notch, which I'll be talking about today. So again, it's at the Fremont Weir. And so the idea essentially is to create a large notch in the weir and replace it with a few gates that would be able to convey some of those waters into the yellow bypass on a more regular basis to recreate that juvenile floodplain habitat and create adult fish passage on a more regular basis than what's available now, which is really only during those overtopping or during those flooding events. So what would this actually look like? This is an aerial image taken a few years ago at the Fremont Weir. And then here is an aerial rendering or rendering of what the project itself would look like. So orienting you a little bit, we're looking to the south here with the Sacramento River running from right to left across our screen. In the middle, we have the Fremont Weir. Out beyond, we have the Yola Bypass, and essentially water would flow from the Sacramento River into the project through these gates. And then from there, it's able to spread out onto the landscape downstream throughout the Yola Bypass area. Zooming in a little bit more, so now we're looking to the north of the Sacramento River in the background. There are 3 different gates that water would be conveyed through. The first gate is 18 feet tall. The second and third gates are each 14 feet tall with a cumulative width of around 90 feet. The structure would be able to be operated from November 1st to March 15th every year and convey up to 6,000 CFS or cubic feet per second of water passing through this structure. Zooming in one more time, we see that this is what the gates actually look like when they're in the closed position. Again, this is a rendering, but we have 2 shield plates that are together, and you can see in this the profile view in the upper right that there's their bottom hinge bladder controlled gate. So when the bladder is inflated, the gate is up, and then the bladder deflates and the gate goes down, which allows fish to volitionally pass through this area and get to the other side, to the Sacramento River. So next up, I'll talk about where we actually are in this process, what's been done thus far. So beginning in June 2022, there was a groundbreaking ceremony with representatives from a number of different agencies, many of which, again, are here today. Following that, there's quite a bit of channel excavation that took place. So this is looking downstream from that gated structure. So there's quite a bit of earth moving that needed to take place, as well as rock placement throughout that downstream channel. After that, there was quite a bit of concrete placement that took place at that gated structure to provide that foundation. So this is kind of earlier in the concrete placement, and this is a little bit further along, where we see quite a bit of progress and a bridge deck being in place. After that has been the process of gate installation and testing. And you can see that one gate in Gateway 1 is in the upright position, again, where it can go up and down. And then that is kind of the current status now. This is what it looked like during one of those overtopping events or during one of those flooding events. And so the estimated project completion is in fall 2025. So this year, there have been a few delays with gate testing and also with this overtopping that I've mentioned. The Fremont Weir is flooded 6 separate times during the course of this construction. So there are just difficulties that come with working inherently in a floodplain. And so that is where we are at currently. But what's the importance of that? So I want to talk next about like why this actually matters and what we'll be doing in the future. And that is adaptive management. So with this structure in place and this way to improve habitat and passage within the Yola Bypass, it's important to have a way to monitor project effectiveness. So therefore, we've been planning a series of adaptive management studies that can help us to evaluate how well the project is working and see what changes might be needed moving into the future. And so there have been a number of plans that are in place. One is to collect air sonar imagery for fish passage in the region. Another is to monitor juvenile salmon routing, passing for juvenile salmon that routes into the structure and pass downstream. Another one is looking at downstream stage monitoring within the Yola bypass. And then the final one, or another study is looking at adult fall run Chinook salmon acoustic telemetry. And again, there's a number of studies here. Please feel free to reach out with questions about any, but today I'll be talking about the adult salmon acoustic telemetry study, which has been mentioned earlier in the white sturgeon talks, but essentially what the acoustic telemetry does is we will catch fish out in the wild. So we'll catch fish in the Yola bypass. We'll affix them with an acoustic transmitter that sends out a unique ping. It's picked up by a receiver, which is the device on the far right of the screen. And so we see that for a fixed receiver, a fish will swim by that point. We know that that specific fish passed that point at a specific date and time. And so in that way, we're able to track fish movement throughout the Yola bypass and potentially beyond as well. And we do this study for fall run Chinook salmon, which helps us to better understand movements of endangered winter run and threatened spring run Chinook salmon as well. So this study has been going on in some capacity in the Yola bypass since about 2012. And it's a huge collaboration with the DWR Aquatic Ecology Unit, plus a number of DWR and external field crew volunteers. This is an aerial video taken by our DWR DICE UAV team that shows our gill netting setup that we have, and we see a salmon that was able to be captured here. And so we use this plus also a fyke trap to catch fish that we can then tag. And we did quite well this last year. So we caught and tagged a total of 42 fall run Chinook salmon this past fall. And then next up, I'll talk about the immediate results from that. So looking at the acoustic telemetry data and seeing where did these fish go? Where do they go within the bypass and why does it matter? So this map has a bunch of red dots for points where we have acoustic telemetry receivers within and nearby the Yola bypass. And that blue triangle is where we tag our fish. And then this table, I'm going to show the number of fish that we see in each of these regions, where we detect them for the last time. So here we're looking at the northern bypass, and we're going to look at the number of fish that we saw for the last time in the northern part of the bypass. So when we tag our fish, and if they go to the northern part of the Yola bypass, they could take a track, something like that. They could go to Wallace Weir Fish Rescue Facility, where they can then be brought back to the river. If you want to learn more about that, you can see Brandy Smith's poster in the session this afternoon. So this can be a beneficial outcome, but it's also possible that fish that go north get lost somewhere within this region, or in certain times they can get out of this region via overtopping, but often not during fall when we're tagging them. This year, we saw 13 of our fish were seen for the last time in the northern Yola bypass. That's 13 of our 42 fish that we tagged. Next up, fish can go up Puda Creek. And so this is a really positive outcome. Puda Creek has been the subject of quite a bit of restoration work over the past few decades. And there's quite a bit of spawning habitat up there. And so they can go from our spawning or from our capture location up to the Puda Creek spawning grounds. And this year we saw that 16 of our tagged fish, so 16 of 42, were seen for the last time up Puda Creek, so quite a few that went that way. Next up, they can end up in the central Yola bypass. We can see them for the last time in the central Yola bypass, where it's possible that they get lost and just end up in this part of the system. It's also possible that the acoustic tags that we affix get shed in some capacity, or that mortalities take place. This year, we didn't see a ton, but so there are 5 of our 42 salmon that were seen for the last time in the central part of the bypass. And then finally, they can go to the south. And so this is an exit point, so they can swim to the south and then exit the Yola bypass and then swim up the Sacramento River or further up from there to the American River, or they can go further to the south. And this year we saw that 8 of our salmon were seen for the last time in the southern part of the Yola bypass. So quite a bit of spread throughout these different regions. And next up, I want to talk about how this might change with Big Notch in place again. So Big Notch can be starting this year in 2025, fall of 2025, and it can open beginning November 1st. So that means the flow can start entering the Yellow Bypass beginning November 1st. And this year in 2024, 31 of our 42 tagged salmon were still within that central part of the Yellow Bypass come November 5th, which is during the time period when flow could start entering the bypass. So that means that a lot of, again, fall run Chinook salmon are in that area still, and so they could have an exit route available starting this next year. And that's even more so the case with late fall run, winter run, and spring run Chinook salmon as well. So how do we expect these numbers to change in 2025? We expect to see more salmon seen for the last time in the northern part of the Yellow Bypass with Big Notch in place, just because that migratory corridor will be available to them and they can go through the notch and into the Sacramento River. For Poudre Creek, we should expect numbers to probably stay fairly similar in that Poudre Creek will have flow before Big Notch will be in its operational season. And so we still expect to see a decent number of fall run Chinook salmon going up Poudre Creek to the spawning grounds there. For the central bypass and the southern bypass, though, we expect numbers to probably drop. So we probably won't be seeing quite as many adult Chinook salmon ending up for the last time down there, just because they'll be able to move to the north because that migratory route will be available. And then getting into implications, as I just mentioned, this new migratory route will be available for these adult Chinook salmon beginning in 2025, but that's also the case for adult sturgeon and other adult upstream migrating fish. Similarly, there will be floodplain inundation available within the Yellow Bypass starting fairly shortly as well. So again, in 2025, when that flow is able to pass downstream, that juvenile salmonid rearing habitat will be more readily available. Another implication is that it kind of creates a change in the hydrology of the Delta region. And while Big Notch itself is on the Fremont Weir, which is outside of the legal Delta, the Yellow Bypass itself is not outside of the legal Delta, and there'll be more water, more available in the Yellow Bypass in upcoming years, and that could have impacts as well for restoration sites, such as what we just heard about from Dan's talk as well. Another implication that is, again, as I mentioned, this acoustic telemetry study will keep on going, as will a number of other studies, and so there'll be quite a bit of data coming along shortly. So please do stay tuned as some of this work gets rolling. There have been hundreds of collaborators really across DWR and different IEP agencies for both the Big Notch project and for the acoustic telemetry studies. So a big thank you to lots of folks in the crowd here today. And please feel free to reach out to me or to this Big Notch email alias for any project specific questions that may come up. And with that, thank you all very much for your time.
We have about 2 minutes for questions. Any questions online?
Yeah, we've got a couple online, actually. I'll start with just one. How much do you think poaching is contributing to the adult salmon going missing in the Yellow Bypass?
Yeah, that's a good question about poaching levels. And so there's probably some poaching that does take place, again, with the salmon closure, the salmon season closure for these last couple of years. Theoretically, numbers should be decreasing. I think with any of our tagged fish, it's probably fairly unlikely. Just we had 42 tagged salmon in the system, where I think on the order of a few hundred have gone up to Wallace Weir this year. So the likelihood for any one of our given fish, given one of our fish is probably fairly low, but that is something to look at as well.
Do you have another one? Is there any sturgeon monitoring plan for the Big Notch facility?
Yeah, so that's a great question. So yes, there's sturgeon monitoring planned. I wasn't able to get to it, but we do lead a sturgeon tagging study, acoustic telemetry tagging study within the lower Yellow Bypass, so we can see movements within the area. The ARIS sonar monitoring is also able to detect sturgeon movement. So we have that in place for another facility in the area, and that will be adapted for Big Notch as well to see successful upstream migration. I think we have one more as well at this time.
Is it a quick one? It's 2:30.
Yeah, great talk. I was just curious about the window from November 1st through March 15th. Is that adaptable? Are you planning on kind of assessing how it goes with that and changing it perhaps, or?
Yeah, so good question. So that is fixed. So that those dates came about from negotiations with landowners further downstream that are planting rice, say within the Yellow Bypass, like the need a dry out period after March 15th, which is the end of that period. There could be some small amount of flows that would be that could be passed between March 15th and through May, but it couldn't exceed the canal capacity further downstream. Yeah, good question though. Thank you all.
Our final speaker for this session is Joseph Merce with Kramer Fish Sciences.
Thank you. Really appreciate you all being here with me this afternoon. Let's just jump right into it. I'm going to share with you some of our results from low impact monitoring at Dutch Slough. Dutch Slough is a really neat restoration project. One of the largest in the estuary to date, about 1,200 acres of farmland being reclaimed or rehabilitated to tidal marsh and channel habitat. Some of the key goals for the project are to restore habitat for native Bay Delta species, primarily fish, support aquatic food web, and contribute to our scientific understanding of habitat restoration. So if you take a look here, kind of always reminds me of loans, right? This really cool project. You've got two parcels, Emerson on the left, which is the larger of the tidal channels, Gilbert on the right, and you can see that there's different elevations of wetland habitat. You also see that big, large blue area there. That's a borrow pit that was used to bring the soils up to get tidal habitat to happen. That's now almost 30 feet deep in the center of it. Another really interesting part of this project was the fact that a lot of effort was put into revegetating the site with native vegetation, and then was allowed to establish for about 2 years before the levees were breached to facilitate tidal action. Some key monitoring issues related to the project are the fact that fish utilize a variety of habitats in space and time, and we've got a lot of listed species. So it really articulates the idea that we need standardized monitoring across ecotypes, but we also need to try to reduce handling of organisms, which is really the focus of our work. So we set up a BARI type study, basically sampling before and after the levee breaches. We sampled with this platform, which is a flow-through system. We collected water quality, primary productivity, well, chlorophyll A is a surrogate for primary productivity, secondary productivity, which was zooplankton images, and then fish we monitor with video and eDNA. So some of our key hypotheses were that new tidal features provide, Joe's got to break out the glasses, wow. Okay, new tidal features provide fish habitat. Native fish utilize those habitats. Habitat complexity affects water quality, and therefore residence time of water, ergo the aquatic food web. And then the aquatic food web evolves over time. And then of course, climate variation over time is going to confound our results. So we broke the features up into sampling open water or center channel habitat and nearshore habitat, and also sampled in our reference locations. For zooplankton, we used a deep focus particle imager and basically set up a deep neural network, basically machine learning, to build a classification model for 3 taxonomic groups. And that's the copepods, the cladocerans, and amphipods. Also in 2021, we really started advancing our machine learning for identifying fish and tracking them. And by being able to count these fish, and notice the confidence intervals there for the machine learning, we can then come up with an estimate of catch per unit effort. We also collected eDNA with transects on the sampling platform. We also did surgical strikes with kayaks. We tried to sample typical on an outgoing tide to avoid exogenous eDNA in the samples. And then we compared DNA sequencing with voucher specimens. So our results. Our sampling regime pre-breach, we made 237 transects before the breach happened that one year before, and almost 700 samples in the following 2 years. Let your eyes catch onto this. There's a lot of information going on here. The upper right-hand corner is that kind of caricature. Notice the color coding to each one of the features, and then it corresponds to the lines, color-coded lines. We're looking at water quality here. The black line there indicates to the left being pre-breach and to the right being post-breach. And then also notice on the bottom side there, we've got statewide precipitation, the percent of average. So the pre-breach, we were in a critically dry year. The year after, we had about 76% of normal. It's kind of silly to say in California. And then 41% the following year. And what we can see is, for instance, with dissolved oxygen, Emerson and Gilbert, which are the 2 tidal channels, their DOs were pretty friggin' low compared to the reference locations and that open borrow pit. And that's probably because that soil, that wetland soil, that interaction, they're probably keeping DO quite low, not to mention those channels were vegetated, so really protected from wind. On the flip side, the deep borrow, big open area, most of the soil gone. And you can see the dissolved oxygen was higher. We also see generally lower dissolved oxygen in the new channels. Again, primarily, I would assume because of the vegetation along the banks. And it really highlights here that design had an effect on water quality at these sites. Moving on from that to primary productivity, design and climate matter. Turbidity on the left, chlorophyll on the right for the Emerson tidal, and then the deep borrow just for some comparisons. And what you can see here is that pre-breach turbidity was relatively low and primary productivity high. And think about that for a second. That there's an infinite residence time of water almost, right? Because the breaches hadn't happened yet. So turbidity is going to drop out. Primary productivity is going to go up. Once we did the breach, now you've got tidal influence twice a day hanging on, well, basically washing out. So you've got turbidity going up, primary productivity going down. But because of the vegetation along the edges, its action is friction or roughness, so it's hanging on to primary productivity. Continuing on with this idea of environment designing of these features, this is an environmental gradient of water quality and primary productivity. So did a snapshot from the spring immediately after the breach and then the following year in the fall. And what we can see is that habitat complexity, so tidal wetland elevation, affects the water residence time and water quality, ergo the aquatic food web. And primary productivity was significantly higher in the mid-marsh versus the low marsh. So again, design matters. Moving on to habitat type and zooplankton, we can see, first off, note the scale. Again, before we've got no tidal influence, that water is sitting there. The zooplankton numbers are through the roof compared to post when you've got tidal flushing twice a day. So we've got the marsh type significantly affecting zooplankton densities, and then a flip-flop from open water zooplankton having the highest CPUE pre-breach to the shore, near shore, and the vegetation having higher zooplankton post. Moving on to the differences between the three taxa, you can see near shore to open water comparisons here before and after the breach. We've got taxonomic differences between near shore and open water and a general positive correlation between density and the distance from the breach. Moving on to early life history of fish. The green is after the breach, above or before is before the breach, as you can imagine. Catch breeding effort was higher near shore, and catch breeding effort increased with distance from the breach in the tidal channels. We're kind of seeing a pattern going on here, right? Habitat size and complexity appeared to affect densities and demonstrated design value, and the results suggest that the longer sloughs benefit early life history by increased water residence time, wind protection from the vegetated banks, and increased primary productivity. And the higher larval fish densities near shore and mid-marsh features again articulate that these designs had meaning. Moving on to juvenile and adult fish, grouping those larval fish, those are fish that are smaller than about 16 millimeters. These are everything that was larger than that. Well, in the new channel, catch breeding effort was generally lower than the reference size pre-breach, but notice that big green line there, and that's for Gilbert, and I'll show you a video in a second from that. Catch breeding effort increased post-breach, becoming relatively comparable to the reference sites. And here we are with a shot from Gilbert before the breach. Okay, we had everything from vector control dropping mosquito fish in to local anglers dropping bass in. These are young of the year that were produced before the breach actually happened. So we had a functioning aquatic ecosystem even before the breaching occurred at the site. With that in mind, habitat effects really did have effects on juvenile adult fish. We had higher CPUE near shore and higher mid than lower marsh. Again, we're kind of seeing a pattern here, right? And while the overall densities weren't different compared to our reference locations and the new sites, think about this for a second. By creating all this complexity, we've got bigger surface area than to mass ratio in these new channels than open water. So actually the carrying capacity for these channels is higher, if that makes sense to you. All right, this articulates overall fisheries benefits by shoreline complexity addition to the restoration designs. Moving on to species richness. Overall, we had an increase in total taxa, fish taxa, documented both by video and by eDNA across all new channels for the two post-breach years. The differences were primarily driven by non-natives, unfortunately, and this was also documented by eDNA and video, with much higher species diversity detected by the eDNA. With native fish, we saw increased native fish species across all sites and monitoring years, excuse me, and higher eDNA signals from native fish like targeted sacramental splittail and Chinook salmon being detected in those newly constructed sites, which really suggested that they utilized these new features. All right, a little bit messy here, but just let the greens coloration guide your eyes, okay? To the left is native fish, to right is non-native fish. And notice the reference locations within each one of those tables and then the new sites. And what we can see with the native fish on the left side is that green is basically virtually all samples that were taken, the eDNA of that species was detected, and white means no detection, okay? And we can see that hitch, Sacramento pike minnow, split tail, sculpin, Thule perch, all fairly consistently seen in the reference and in the new locations. But look at those friggin' non-native fish. They were everywhere. We had black bass, we had Mississippi silversides, we had gambusia. It was insane the number of non-native fish that were out there, both in the reference and in the new locations. Now, I really like what our genetics lab did here. This is a dissimilarity matrix with basically what species are pulling the sites into different locations. Let your eyes grab onto that gray and dark blue. That's pre and post breach in our reference locations. Note they're basically the same thing. As you would expect, the reference locations out in the Delta, the breach should have no effect on that, right? But check out the ones that are over in the light blue, that light grayish area, that light blue, the mustard color, the green. Those are our new sites pre-breach, and they basically shift to our reference locations post-breach. The last thing with diversity here is using eDNA, looking at average read counts. For the number of samples that were taken that were read for eDNA, the circles are relative to the amount of times that species was picked up there. And we can see a couple of cool things here. The yellow on the left-hand side is pre-breach, so we had some native fish like the tule perch, sculpin, and the hitch being in the new locations before the breaching occurred. But check out all the new species, their fingerprints being picked up afterwards. Cool things here is, number one, we're seeing an increase in numbers, but the second thing is note the seasonal variability for some of these species, like the split tail, right? And remember, 2023, a very wet year. Look at, for instance, split tail and how consistent it was seen in the wet year versus the first breaching in a relatively dry year. So we're actually picking up seasonal variability of detections of these fish out there. So management implications. Design tidal channels support aquatic food web production and increase physical habitat. Tidal channels evolve over a relatively short period, but climate makes it complicated. The channel design and wetland interactions should be considered when planning tidal wetland restoration. Furthermore, while native fish species were increased over time and the sites were dominated by non-native fish. So habitat restoration alone is not going to fix all the woes of our native fish fairly clearly. Even so, designed high complexity tidal habitat appears to be more successful than haphazard levee removal. Future directions, we need to update the definition of Bay Delta fisheries restoration. It's really important to see here, there's a ton of really cool monitoring going on out at Dutch Sloop, and this is only one facet of it. There are folks looking at sediment, soil development, carbon storage, terrestrial vegetation, birds, insects. There's some really cool stuff going on here. We shouldn't just be fish focused. There's a lot of really wonderful stuff going on out this site, and we got to keep an eye on that. We also are our group ourselves are really going to continue to work on improving upon image analysis and eDNA quantification. And with that, I'd like to acknowledge and show appreciation to the Dutch Slough Research Working Group, the dedicated field and laboratory team that helped us get this all together, DWR staff for not only helping us do this and design it, but funding it, and then IEP for the collaboration and permitting support. And with that, if you have any questions, I would love to talk to you about them.
We've got about 3 minutes for questions. Anybody online?
Yeah, we've got one online from Tyler. Was there any noticeable change in fish size after breaching?
You know, there's quite a bit of seasonal adjustment. So again, we were seeing young of the year bass and we were seeing increases in size. So yes, we did see differences, but to say, did we see smaller fish before the restoration and larger fish after a habitat restoration effect? No, we wouldn't be able to detect that. That would be too small of a scale, I think.
Hi, really great talk. I noticed that there was a substantial decline in zooplankton density post restoration. And I believe in your explanation that because of breaching, there's lower residence time. But given that pattern, what would you measure to show that there's a success in restoration when it comes to food productivity?
So a couple of things here. One would have to be a calculation of biomass produced, right? So not only standing crop, but how much is actually being exported out of the site. That would be far beyond what our techniques would be able to do. So that would be the next step. But the fact that we were seeing higher primary productivity on the near shore in the vegetation than in the reference locations, I think is a good enough articulation that the sites are having a positive influence. The last thing you got to think about here is the fact that you're actually increasing the surface area of water out at the site. So even if you don't see a significant increase, overall, you're actually increasing the carrying capacity at the site. You bet.
Thanks, Joe. That was really interesting. Yeah, one of the things I think with residence time, you see larger residence time when you have like a bigger body of water. Is that somehow designed into this, or is it mostly these kind of capillary channels?
You know, again, the design, I got to hand it to the PWA ESA crew for designing this. It really, I mean, it's a pretty beautiful site, right? I think they really were trying to design features that were going to maintain themselves over time. So there's a dance between trying to increase surface area versus actually having this function like a tidal feature. So I was not part of the design team, and I can't tell you any more than that. I will tell you that big, giant, open borrow pit, there were times that thing is producing some primary productivity. Yeah, definitely. Yeah. All right. Thank you.
Thanks, everyone. That concludes session 4. Up next, we have the Poetry Slam.
All right, folks. I hope that it's not just me up here with a poem. If you have brought a poem, skit, song, rap that you would like to share, if you can come up front and just line up to my right, we can get going. Please don't leave me hanging. If you're all exiting the room because you're scared of poetry, I'm outing you right now. All right, well, here is my contribution. Again, we all pack an arena to talk about science. We've seen a few talks about fish over coffee we dish, and we put PowerPoints up on a screen.
Hello, I'm Trevor from the USGS. I hope you'll come visit my poster. GSSMB, Affect Hydrodynamics, Bubble Barrier.
Hi, Michelle Nelson, DWR. All the credit goes to Rosie for this. All right, this is about my poster. There once was a bay we called Grizzly, big enough to make your hair go frizzly. We put out some buoys whose data were screwy. Other stations were similar for shizzly.
Hi, I'm Katie Hostetler with IEP, and I have a poster, and this one's kind of long, but here we go. Beneath the waves so small and seen, zooplankton drift where tides convene. A flowcam snaps their fleeting trace, a data set born in time and space. With cultured care, they thrive and grow, captured in the scope's glow. A library builds each shape, each hue. The model learns what's false, what's true. Ecotaxa, sharp and wise, sees through the plankton's disguise. A test, a split, a model's might, 0.98, our path is bright. From pump to pixel, lab to code, science walks where data flowed. Machine and nature side by side unravel the mysteries of the tide.
Hi, I'm Margot, and this is a advertisement for the Delta ISB's food web poster upstairs. The Delta, where two rivers intertwine, organisms are linked by food web lines. but the upper trophic crew needs research anew to help us refine a food web model design.
Okay, we didn't have a ton of people last year either. The fact that we had a small but mighty crew reading poetry today may mean that I have lost the fight to revive the Poetry Slam, but it's still a great time for those of us who shared. Thank you very much. We're now gonna go to the poster session. The poster session is up the stairs and over. There's signs, you can't miss it. If you need any assistance getting up the stairs and need the use of an elevator, see Christine, Job in the front. The poster session will be just for the rest of the day, going on till about 5:30. This room is gonna get closed up, so please take everything with you as you go upstairs. If this is the only day you'll be at the workshop, please deposit your badges on your way out. And the other reminders I have are, there is a Cal Neva sponsored happy hour at Drake's the Barn, starting at 5:30. And I think Pete wanted to make an announcement about tomorrow's after hours event.
So for folks interested in climbing at Pipeworks tomorrow, everybody is welcome. Beginner, experienced, doesn't matter. I do have some volunteers who are going to help me serve as belayers and you know, showing you around if you're new at this. But if there's anybody experienced who has a belay card there, or you know, doesn't mind getting one and would be game to help out, that would be great. I've got a few, but a couple more certainly couldn't hurt. Just come find me today up at the poster session or sometime around here tomorrow, and I'll clue you in. Thanks.
All right, thanks everyone. I'll release you to the poster session and we won't be having a final wrap up. So I'll see you tomorrow at 8:30, if not before.